seminar. My name is Joe Ross. I'm a physician and health services researcher at Yale in the School of Medicine and the School of Public Health. And along with Molly Jeffery and a whole team of amazing folks, we lead the Yale-Mayo Clinic CERSI. The CERSI is an extraordinary opportunity for those of us who work in academic settings to have the chance to work with people who work in government at the FDA to try to work together to address key gaps in knowledge through research and develop tools to support regulatory decision-making, addressing questions that the agency may have where our academic skills and mission may be able to supplement and enhance the work being done at the agency. The Yale-Mayo Clinic CERSI is one of 5 CERSIs that's funded and supported by the FDA. We're now in year 9 of this joint venture. There are also CERSIs based at the Johns Hopkins University. There's a joint CERSI at UCSF and Stanford. There's one at the University of Maryland, and there's now a Triangle CERSI that's now, I think, going into year 2 that includes the University of North Carolina, Chapel Hill, Duke, North Carolina Central University, and North Carolina State University. We have now, I think this is our 4th year running the Scholar program, where we have trainees at Yale and the Mayo Clinic, medical students, master's and PhD students, and fellows on our campuses engaged in regulatory science who've applied for and been identified for doing relevant and excellent work to participate in this program. And it sort of culminates in this scholarship virtual day at the end of the year. We also invite similar level trainees from each of the 4 CERSIs, along with the Harvard MIT Center for Regulatory Science to present. And so there'll be 12 individuals here presenting today on their work. We're excited to hear what they've been up to. Maybe the projects aren't yet finished. That's okay. There's still benefit from feedback, and we're excited to learn what people have been involved with. And we're honored today to have Stephen Kozlowski to give a keynote as part of this event. Dr. Kozlowski is the FDA's acting chief scientist, and in his role, he promotes, leverages, and leads cross-cutting collaborative activities and initiatives that catalyze FDA science, innovation, and research to help the agency address its most pressing regulatory and public health questions and respond to emergency issues. Prior to assuming the role of acting chief scientist, Dr. Kozlowski was responsible for ensuring the quality of all the active ingredients and biological products overseen by CDER in the Office of Pharmaceutical Quality. He holds a bachelor's and medical degree from Northwestern University and trained in pediatrics at the University of Illinois. Dr. Kozlowski, thank you for joining us. All of our trainees are eager to hear from you today, and we appreciate the time in your busy schedule to come and meet with us and kick off our event.
Well, thank you very much, Dr. Ross, for inviting me. I am going to share my screen. Okay, are you able to see that?
Yes, we are. Thank you.
Okay, great. So I'm going to talk about regulatory science for a slide or 2, and then about one area in it, advancing alternative methods. And so I want to go through definitions of science. So I think we all value, and probably in some ways the most important science is fundamental or basic science. You think about non-Euclidean geometry waiting so long to find its place in general relativity that science for its own sake has tremendous value, but it is not the only kind of science. There's also applied research where the science is directed for more tangible goals. Translational science where we leverage the amazing fundamental science knowledge that has been generated and figure out how to turn that into practical, tangible outcomes for people. And a subset of this is regulatory science. And regulatory science deals with what we need to know to make sure that these new ideas, innovations, tools are really safe and ready for the public, that they can be controlled to make sure they perform the way they should. And so it's a very important subset of science that allows all these other branches of science to be leveraged. Dr. Ross talked about CERSI, talk a little bit about it. Basically, the CERSIs between FDA and academic institutions really focus on regulatory science. It's in the name, Centers of Excellence in Regulatory Science and Innovation, and in a variety of ways. Research with FDA subject matter experts, visiting scientist programs, workshops, exchanges between CERSIs. As was mentioned, there are members of different CERSIs who are involved in some of these scholar programs. Training programs, and other fellowships. The Office of Research and Emerging Sciences, part of the Office of the Chief Scientist, really supports and makes sure that these can work. Centers and offices outside of that group really fund and work on regulatory projects within that group. And as mentioned, there's a 5-year agreement and there's a rigorous process to assure the quality of the work being done. So I want to jump back to regulatory science. So recently, not that recently, but there was a report on focus areas of regulatory science. And as you can see, there are lots of items. I'm not expecting everybody to read every item, but I want to highlight one: novel technologies to improve predictivity of non-clinical studies and replace, reduce, and refine reliance on animal testing. And that's what I want to talk about a little bit today. Now, I do want to note that the category in which that particular area lives is increasing choice and competition through innovation. And innovation also lives in the eye of CERSI. So I want to take a moment to think a little bit about what we mean by innovation. So a definition, this is from Wired magazine, right? Innovation is not invention. It is not creating something new alone. That's invention. But innovation is an invention that actually makes a difference. It's being used. It changes interactions. It changes behaviors. And so it has an impact. And so there's a lot of thought about what makes innovation, and in fact, what is the most powerful innovation? So a term decades ago that was brought up and still used frequently today is the concept of disruptive innovation. So a disruptive innovation is innovation that creates new market and value networks and disrupts an existing market and value network. So those are very business terms, but it really means that the way people do things, the marketplace, changes in a significant way. And the term was defined by Clayton Christensen in 1995. And one thing that's really important in the definition of this is it's not a zero-sum game. We think about, digital photography and Kodak leaves. It doesn't have to be that. It can be really impactful changes, even if it doesn't completely disrupt the providers and the systems. It's really about significant impact. And I think the question always is pharmaceutical development, are there things that can be, whether they're disruptive of the whole system or just highly impactful, what things can really impact pharmaceutical development? So in this early discussion of what makes disruptive innovation, the first thing, or one of the key things, is the technology. There has to be something that enables some benefit. There has to be a way of translating that into a marketable business with cost recovery, with some system of being able to operate in that. There has to be outside groups who want it, whether it's customers, patients. Somebody needs to really see value in this. And then last, but maybe not least in this context, is there need to be regulations and standards that allow all of these to work together in a way that's safe and effective and meets standards. So example, if you were replacing some form of safety study, like animal studies, you would need to assure safety in the new way of doing it, but you would also need to assure it in a way that doesn't create unnecessary blocks or hindrances, right? So the standards and regulations really need to be thoughtful and careful. So to go back to this area we're talking about, technologies to improve predictivity of methods that don't involve animals, what have we done so far in this area? What's been invested in driving this outcome? So this has actually been a very long-standing effort. Um, you know, well over a decade ago, the FDA generated documents about modernizing toxicology. We've had a roadmap about toxicology in 2017. In 2021, we had a document about alternative methods. We have had a myriad of cross-agency working groups on toxicology, alternative methods, actually two of them on alternative methods, including a group on modeling and simulation. We have promoted regulatory science research, and we've had a variety of collaborations, and I'll just mention a couple with NIH. We had MOUs with multiple partners at NIH on advancing toxicity testing, and between NCATS and us on microphysiological systems. So this is a long-standing effort with lots of investment. A little more recently, in 2023, in the FDA Modernization Act, Congress actually defined the term non-clinical test and allowed it to include results from alternative methods to animal testing. So there's actually now a statutory basis for doing this. And they invested money. There was $5 million in core funding in 2023, more in 2024, to drive new alternative methods and to spur its use, streamline development of FDA products, regulated products, bring things to the public more rapidly, and really assure that the products are safe and effective and patients can depend on them using alternative methods. So we have also generated a variety of guidance documents to give industry a sense of how to do this. There are guidances on specific areas of safety, which can touch upon this. There's oncology and photo safety. There's guidances on specific alternative methods, computational methods, one of these guidance documents does. And there's guidance on the qualification process. And so I want to talk a little bit about the qualifications process. So in 2016, in a bill called the Cures Act, Congress created a pathway to qualify drug development tools. And this allows for transparency and a very fixed process in qualifying these tools. So what does this mean and why? So before this pathway, medical product developers could always submit data for an alternative method in their application. They can always say, I have a better way of doing this, and the agency evaluates it. But it's kind of a risk for companies because you want to get your drug approved. There's a lot depending on it. If you have to depend on a parallel evaluation where it's new to reviewers and they might say, wait a minute, what about this? You're kind of putting your product at risk. So having a separate pathway has value. And also it allows for having a very specific context of use for something that might be more generalizable than the application of one product. And so combined with that transparency piece, it may really allow the benefits of one alternative method to really be leveraged by more companies, maybe by more products within a company to really help with that. So again, this allows this sort of parallel pathway with a more generalizable outcome. And I think that's why this qualification pathway is so valuable. Now, it's been translated in different centers. Devices has a pathway, again, with its own guidance. Drug developments, both drugs and biologics, have this pathway. And I want to talk about one of them, which is called iSTAND, Innovative Science and Technology for New Drugs. So this is where some of these new in vitro systems might end up being looked at. So till basically this past month, there were 55 submissions to this program. There were all kinds of things. There was digital health technologies, that's a DHT, algorithms, and about a third of them were microphysiological systems or other complex in vitro models. There was a fair amount of publicity in September of last year that the program accepted the first organ on a chip technology as a letter of intent. And I will tell you, a letter of intent doesn't mean it's qualified, but it's that first step in being qualified. So out of all these submissions, 8 of them have letters of intent. Some of them also relate to toxicity, although not microphysiological systems per se. Some of these are moving towards qualification, but none of them are actually fully qualified. Now, there are alternative methods that have been used for a while for safety testing for things like ocular irritation, skin irritation, phototoxicity. There have been alternative tools that aren't used alone, but are part of weight of evidence with other studies. So you can say there's a lot of forward movement, but you could also say, how much have we really moved the needle on animal use? Because there's still a massive amount of animal use in drug development. So I want to talk about the context of change in a more general way. And I'm going to tell a parable which Mike Kopcha, a mentor and boss of mine, has used. And I think it's a bit of a metaphor, but it's about bamboo. And the idea is you might plant bamboo and it will take a long time. You know, the metaphor goes years. I don't know scientifically if that's necessarily totally, demonstratable, but that there's a long period of time in which the seed is sprouting underground where it may need watering and care, even though there is nothing visible to be seen. Yet, when the sprout appears, it actually may grow very, very rapidly, sometimes to very large heights in the matter of weeks. And so this parable is kind of at the concept that there's a lot of work that may need to happen for an outcome, and that outcome may not be linear. It may be that that work occurs, and then at some point, there is a large change in outcomes. Kind of an example about that are monoclonal antibodies. Um, you know, they were published in 1975. There was one product in 1986, OKT3. And then for close to more than a decade, there was nothing else. And it was really until more than a decade later when suddenly this area where now there are well over 150, that's probably a huge underestimate of monoclonal antibody products. And that was driven by molecular biology, where engineering antibodies that were chimeric with mouse and human components or fully humanized, except for the complementary determining region. And so this wonderful technology sat, and there was other things going on. And then when it coalesced, suddenly there was an opportunity for real growth. And I want to take this back to animal models, and I want to talk about biosimilar biological products as an example. So biosimilars leverage information from a reference product, right? They have a standard, highly similar, no clinically meaningful differences. And the statute says use 3 types of data to make that decision. Analytical studies, animal studies explicitly, and a clinical study or studies. From the beginning, there was a lot of view that the animal studies were not necessary. You already have a molecule that you're showing is highly structurally similar to another molecule. At what point does these animal models really add more information than what you already have? So again, that percolated for a long time. And in 2022, the Fedora bill was passed, and it changed the language of the statute. It basically said that instead of animal studies, an assessment of toxicity, which may rely on or consist of studies described elsewhere, like the analytical studies may be leveraged for this. The consequences of this were in 2024, there were close to 20 biosimilars approved. Public review data is available for about 3/4 of them. In none of them did the FDA ask for animal studies. Sometimes sponsored volunteered it themselves. I'm not sure why, but yet it, again, a situation where the value of this was described and discussed for a long, long time, and then suddenly, right, there are changes and it really becomes embedded in practice. So how do we drive this more broadly in terms of reducing animal use? What are factors for innovation? So the value of this, I think, in innovation is, 90% of drugs, generally the success rate for a drug once it's an investigational new drug is about 1 in 10. Maybe a third of those failures are purely toxicology. You can debate that, but the vast majority are some combination of safety and efficacy. We know animal testing is wrong. It misses things. There are clear examples of that. And there are always ethical concerns about animal testing and cost issues. So there's certainly a broad network that sees value in this. There's also business case that these could be faster and create greater efficiency in drug development. Human in vitro models may have advantages in some cases over animal models and maybe better prediction in some cases, faster delivery of therapies to patients, and the reduced cost of animal testing. So both from a societal value and a business model, I think those factors are definitely already in place, and potential cost in drug development savings, which results from all those things. So recently, the FDA released, I think, April 10th, a new call for action on reducing the use of animal models. Here we go. It's April 10th. And it involves a roadmap. And that roadmap really talks about really moving forward on this. So I want to start with this new call to action and take a look at the enabling technology piece of innovation again. So there's a lot of technologies that we have, complex in vitro systems, right, that may vary from cell cultures to much more complex models, in silico tools, ex vivo human tissues, other high throughput screening tools that may involve cells. So there is no limit of tools. And I want to talk a little bit about some of these. So in silico modeling. So I'm going to talk about research that the National Center for Toxicology Research at FDA has done, and this may be reflective of a lot of research. So they have an animal GAN model, which creates a digital twin of rats. I think this actually, and that is based on a lot of learning, machine learning, and they use it to predict 38 clinical pathology measurements. So you're not just looking at one outcome, you're looking at 38 different outcomes by analyzing chemical structure, treatment duration, and dose, and then leveraging a lot of data to be able to make those predictions. Now, that's somewhat useful with FDA data because FDA sees, various aspects of this, but it's missing really important data, which is all the animal failures that never went to the FDA, right? Does it predict those studies? And there is some work with pharmaceutical companies to try and really leverage that. But I think, again, these are amazing concepts, but the question becomes, do you need to model histology? Can we really get a lot more training data to really fine hone these predictions, including the failure data that may or may not be visible to the FDA? So again, a lot of potential in tools like this, but a lot of space potentially to really move forward. Talk a little bit about complex in vitro models. So again, they range in complexity in many, many different ways, from cell cultures to very complex multi-organ systems. And there are all forms of them, whether organoids, micropattern cells, and other things. And they're getting more and more complex. But the questions really remain, do they have the relevant cell types interacting in the right ways? Do they consider metabolites and there are ways to try and deal with that? And how can they inform longer term exposures, right? We really need to better understand the capability and gaps in these powerful systems. And any one system is always going to have gaps. But if you combine these tools together, such as in vitro and silico and other ways of being complementary in those things, can we really improve the predictability? And, what do we still need on the technology side to move forward? So I think this call to arms is really about, are there gaps here, what they are, and what it would take to really, really move forward on them. Now, I want to move to the last part of that triangle, which is regulatory standards. So I've talked a little bit about some of these things. And what I would say is the bamboo seeds have been very, very busy, even if they haven't fully sprouted, right? We had the change in the law. We have multiple guidances on these topics. We have a qualification process with steps that's very well worked out, right? So on one hand, you could say, wow, we've immediately moved forward on this. But on the other hand, you can ask, is there more that we can build on this? Is there a better way of making this happen? Um, and so the roadmap talks about this, right? Are there ways we can further map critical endpoints in measuring toxicology and use cases? Is really there more information that we can bring to bear on this? Can we really advance the way we validate and qualify these things? Are there other opportunities there? Is there more guidance that will really create more clarity and help make this work better? And then, even if there's clear guidance, this is a little bit of a culture change, right? It's a different way of thinking about things. And so, how do we really train both within regulatory agencies and also outside of them that this is happening and how to really make this successful? And then again, we're going to constantly need to monitor how we're doing and iteratively refine, and improve the things that we are doing. So I think again, there are lots of opportunities, and in particular, how we think about justifying a NAM may need to change a bit. And so I want to talk a little bit about the acronym NAMS. So NAMS is a very stable acronym. It's been around for a very long time. But what people spell out with it changes, right? It's always had differences. And one thing to really point out about the new roadmap is it uses the language new approach methodologies, right? And what we've used before, what's in the statute is new alternative methods. And I want to think a little bit about what that change in language implies. I think when we talk about an alternative, we're talking about a one-to-one correspondence, right? I use an animal for this. Does this alternative do exactly what the animal model can do? And that's one particular way of trying to validate or qualify something. An approach is broader language. Is it not about necessarily always a one-to-one correspondence, but a way of looking at the benefit overall, that a whole suite of things may not be measurable in a one-to-one correspondence with an animal model. They may have advantages that are not there. They may have a few disadvantages, but overall, are they better for public health? And so I think that's really a mindset change in naming. And so again, we're moved from new alternative methods to new approach methodologies. Is that just, an outside book cover, or in fact, is that something that implies a broader and different way of thinking about things? I also want to mention that the roadmap has an initial focus on monoclonal antibodies. So it talks about lots of things and expanding this to the future. So I want to take a moment to think about monoclonal antibodies. So they are very, very specific, right? The amazing thing about the immune system, through history has been the incredible specificity that it can generate. So often toxicity is on target because of that exquisite specificity. Furthermore, that exquisite specificity also tends to be species specific. So finding relevant animal models for antibodies is often a challenge, and often you're stuck with non-human primate models or sometimes none at all that really are relevant. So that's one reason that there's a focus on this. There's certainly literature, I'm throwing one paper here, where animal models have been useful in stopping development of antibodies. So one has to be really careful about saying they don't have value, because there are examples where they do, and you really need to think about that. But you can also really try and think about the context, right? Antibodies have different structures, isotypes, mutations. They have different effector functions. They may have different valence, half-lives. They may have features like half-molecule exchange. So to try and understand the history of toxicities in the context of the structure and function of antibodies, and also in the context of the mechanism. Some antibodies have incredibly complex mechanisms with multiple methods of cell killing, right? Other antibodies simply bind a soluble target, right? And they're not in contact with cells. And so those things may really matter in how you think about risk. And so there may really be opportunities to really further stratify and understand the way we look at risks. And antibodies may be a really good way to start with that. I think to get here, technology and standards need to co-evolve. They're kind of separate pieces on that map, but they really need to work together. But what is key, what is the starting point, is we still need to ensure scientific rigor and continuity. As we move towards replacement, and this comes straight out of the new roadmap, FDA must assure that any new method is equal or superior to the animal test it replaces in protecting patients, right? That's the North Star. But nonetheless, there are real opportunities. And I think there's a real potential for interaction between the technology we're looking at and how you regulate it. And clearly we have different guidances for in silico and in vitro, and there's going to be a lot of nuances. But nonetheless, when we really think about the technology, that needs to inform the way we regulate it, and vice versa. The things we need to achieve in regulation really need to inform the way we develop these technologies. And so there needs to be a partnership, right, with collaboration in driving success in this area. So again, I have here gears that these things really need to work together in a smooth way. So there are many examples of this, right? The National Toxicology Program has the ICVAM, which is a coordinating committee on validation of alternative methods. By collaborating with NIH, the VA, other government agencies, we can really pool expertise, share resources and data, and really work on developing NAMs much more effectively. I will mention some other, and there are many government collaborations. So Confirmatory is Complement Animal Research and Experimentation. And this is an NIH program which will create or fund technology development centers that really focus on NAMs in greatest areas of needs. They're going to have a resource coordinating center that will ideally create some public-facing searchable structures on NAMs, and a validation network that's going to really look at how these are validated. And I think that piece is incredibly important because we really need to leverage many partners in figuring out what this future way of validating and qualify will look like and how it assures patient safety and yet is not a bottleneck, right, but an enabler of new and valuable technologies. I'll mention another one. ARPA-H has a program called Catalyst, Computational ADME Toxin Physiology Analysis for Safer Therapeutics. They're looking at in silico methods. And again, they have, they're a different set of resources and bring to bear a somewhat different perspective. And so all of these together with many other collaborations, including working with CERSIs, right, are going to be very, very important in figuring out how to do this right. And I will mention that, this very week, April 29th, NIH released an announcement saying they're prioritizing human-based research technologies, that there may be greater value in microphysiological systems with human cells than animals for a lot of the research that's going on. So this effort is much broader than just necessarily animals in preclinical development for drugs, but really sort of have many, many areas where it's involved. And so joint funding initiatives, shared databases to the extent that's possible, and the united front, including a federal united front, will really help move this forward. So lots of things. How do we really begin to implement this? So the roadmap talks about a strategy, and I think there will be really iterative things about this. So first, what pre-existing data can we really look at? And this is always tricky because it's complicated to ask somebody to share information, right, that they don't necessarily need to share to move their application forward. And yet sponsors probably have lots of data where they use NAMs in product discovery and compare it with animal data that would really be informative about this. Open access repositories. Again, people have to be willing to share how to leverage that, how to make that happen. So all three of these items are really about data that exists, right? It's how to bring it to bear. Then are there shorter term things? Again, the roadmap talks about monoclonal antibodies. Maybe it'll be another area, but we really need to look at low-hanging fruit. Are there some things where we can move forward more rapidly on? And we need to really measure what we're doing, animal usage, changes in toxicity testing. If we're not observing and measuring the impact of these things, it'll be kind of going forward blindly. We really need to understand what's working and what's not working. And then in the longer term, there really is the concept in this roadmap, which is a very exciting goal of making animal studies the exception rather than the norm. There will always be situations, the animal rule, where you can't test something in humans, right? Where animals will play a role. But the idea is, for the most part, to really think about what technologies can replace them. And again, that would mean shifting to a NAM-based default for maybe the IND enabling preclinical safety toxicity testing in many cases. So I want to close. I think we're at a time for change, right? Where maybe that bamboo is sprouting, and depending on how we treat it, fertilize, and nurture it, we can really see a more rapid growth. So we really talked about new approach methodologies, validation. We need to really unstick the gears between technology and its capabilities and regular standards and really advance both of them in synergy. We need to really think about a careful staged implementation plan, leveraging FDA experienced scientific partners and always assuring patient safety. And this will enable innovation that in the long term will improve safety, lower cost, speed development, and really be of benefit to everybody. So I want to thank all of you for your attention. And it kind of as a goal, right? Will we really be disruptive innovation where things change so much that people think, did we really used to do that, right? Did we really do that? So anyway, thank you. And I don't know if there's some question time or how we manage this.
Well, Dr. Kozlowski, that was great. And hopefully people will have a couple of questions. But I thought that was such a great presentation, although I'm not sure about the bamboo analogy. As someone whose mother-in-law planted bamboo in the backyard thinking it was going to be beautiful, and I had to spend multiple weekends trying to pull it out as it overgrew. We have to be careful with bamboo.
Right.
But I thought that
it's a good lesson that we want to grow this right. Yes.
Right.
Yes, absolutely.
But what I love about it for our trainees is it shows just how exciting things are at the FDA in terms of how you embrace sort of emerging methods to evaluate new technologies, but the importance of regulatory science to ensure that those methods are reliable and rigor. I mean, my work most of the work of our CERSI is really not preclinical. It's mostly post-market, thinking about methods like real-world evidence. How can it be leveraged and ensure that it's accurate for the post-market evaluation period, or even stuff around clinical trial data sharing initiatives. But you can see the echoes across the different spaces, and so I think this was a great example for our trainees. Let's see if there are anyone has questions. If anyone has anything they want. Any hands up?
I will, add to what you said about real-world evidence. There is a space for that here, and it's even mentioned in the roadmap, because it could be that there is a lot of data on clinical outcomes and animal studies that has not been fully leveraged, right? And so, to me, there's all these things connect.
Yeah, no, and in fact, we've had some conversations preliminary around like the concept of digital twinning, which sort of relates to this, right? Like, in terms of the predictive modeling and what you can do. Hopefully we won't get to the point where we avoid human testing, but I love the concept of avoiding animal testing when it's not needed.
I see a hand up. I don't know.
Hi, my name is David Holmes. I'm at the Mayo Clinic. I really appreciate your comments and the perspective as you took us through that story of moving to these alternatives. At the end, you started to talk about it a little bit, but I want to go a little bit further in who's ultimately responsible for this validating of these new approaches. Now, you talked about collaboration with, NIH or ARPA, and they're trying to do some of that stuff. But the balance here is that you've got companies that are investing just to get their technology moving, their solution moving forward and improved. Their mission's not to validate new techniques necessarily. And if we leave it to the research community, like the open-ended research community, their job is to publish new ideas, and they're not really driven. So there's someone's got to pull all that together and be ultimately responsible for, these are the alternate strategies that we want to explore very specifically and want to validate and have the cookbook of validation, so that people submitting don't have to also do the entire validation of the technique that they want to use to then validate their technology and get it through regulatory practices. So I don't know if that made sense, but who's going to be the owner of responsibility? Is that the FDA? And if so, are you investing in doing that work yourself, or are you outsourcing it? What does that sort of look like?
So the responsibility for validating the methods are with the sponsor, right? The company that brings a drug to us, if they want to use a new method, then they're responsible for it. The qualification process that I talked a little bit about is a way of trying to make that a little bit of a separate stream, right? And that might involve players that are not necessarily the companies bringing the drugs forward, but presenting a context of use, right? And then that becomes more broadly available. So that's one avenue. There may be parts of industry that are actually very interested in developing these through this process. But it is the sponsor's responsibility to tell the FDA that what they're doing is fit for purpose, right, and protects patients. So the question is, in all these partnerships that exist, is there a way of making sure they connect? Because the NIH Validation Qualification Network may end up producing valuable information on this, but they're not going to be the ones submitting the applications. And so it's a thing about, can they make this available? Can the FDA make clear what it expects, right? And we try and do that with guidance, but that becomes very, very important. What do we actually expect, and how do you do it? And it's hard, and there's a gap. And I do think that gap exists. And the question are not whether there are better ways of bringing people together, better partnerships that will help this.
Yeah, and also to say to David's point, I mean, this has been one of the great virtues of the CERSI, which is that it allows those of us in the academic world to work directly with folks at FDA, sometimes even with industry partners as part of projects to do methods development related work. And yes, of course we want to publish it, and that those are part of the incentives in the academic world, but the impact by working with FDA to inform guidance that later on, as methods are refined, and it is one of the great sort of virtues of the program. So those kinds of opportunities are ones to really take advantage of, and we've had a chance to do a few kind of methods work, development work like that with projects over the years, which have been very beneficial.
Right. No, it's example of partnerships, and those partnerships need to be effective, right? And that's a complicated thing. How do you measure that, right?
Yeah.
But the goal is to drive that.
There's one more question in the chat from Frank Lee, who talked about how there's great presentation, a lot of information, and said that one of the big barriers is getting news out, like the guidance documents and the roadmap, and just asked if you have an approach, or how does FDA think about sharing these larger initiatives with the public? When do they get pulled in? And maybe it's different between the public and the scientific community, and how to leverage their expertise.
Right. So these things are on our website. I think it's a good point, and one that I can actually bring back, that how do we better reach out? Because if part of the goal is engaging partners, right? There's some partners we're going to know about and work with, but how do we share this? And again, there's a press release, but that press release doesn't guarantee that it gets everywhere it needs to go. So I think that's a fair, open question.
Well, Dr. Kozlowski, thank you so much for making the time to kick off our Scholar Seminar today. And you're, of course, welcome to stay as long as I'm sure you're very busy and have lots of other things to do, but thank you.
Well, thank you very, very much for the opportunity, and, great work, and, hope we will interact in the future.
I hope so too. Wonderful.
Thank you.
Well, I'm going to hand the baton over to Molly Jeffery, my partner in crime, leading the Yale-Mayo Clinic CERSI, and I will let her kick off our first session, session 1 of the seminar.
Thank you, Joe. No crimes committed here. And thank you so much, Dr. Kozlowski. We really appreciate your time today, and I really enjoyed hearing your take on disruptive innovation and regulation. I am so delighted to introduce the very first session. This is one of my favorite days of the year where we get to hear about some of these projects that, that trainees are working on that are of great interest directly to the public and to our colleagues at the FDA. So the approach we're going to take is we're going to aim for 8 minutes per presentation, and we'll hold questions till the end. We'll have 10 minutes for questions at the end or whatever's left. Please put your questions in the chat. If you are shy, you can send them directly to me privately, and I will share them. You could also raise your hand and just ask your question at the end. So without further ado, we will start with Anishaa Sivakumar from Yale, presenting on IV hydration spas.
All right. Can everyone see my screen? Okay, perfect. Hi, everyone, and welcome, and thank you so much again for having me here. My name's Anishaa, and I'm looking forward to sharing with you our research on IV hydration spas, state policies, and facility practices in the United States. Okay. So first, what is an IV hydration spa? IV hydration spas are commercial facilities that offer IV infusion therapies to the public. They typically offer hydration solutions alone, but these can be combined with other compounds. And those compounds can be electrolytes or vitamins, but they can even be pharmaceutical products, including GLP-1 agonists or ketorolac. And these can be offered for a variety of claimed benefits. Notably, this industry has been growing quickly according to market research, but regulation appears to remain inconsistent. This is primarily because IV hydration spas are typically treated like compounding pharmacies, which are pharmacies that are exempt from typical standards at the federal level, though they must still compound based on patient-specific prescriptions. In 2013, the Drug Quality and Security Act increased oversight of compounding pharmacies on the federal level through the creation of voluntary outsourcing facilities. But for many IV hydration spas, they actually don't have to register since it's voluntary, and regulation ends up being left to the states. In 2021, following a report of septic shock after a patient received IV infusion therapy from a compounding site, the FDA and the National Association of Boards of Pharmacy shared information regarding safety concerns and the need for regulation of these facilities. Several reports of safety concerns at these facilities reported to the FDA with concerns about unsanitary conditions, improper storage, and failure to meet staffing requirements. So with all of this information in mind, our objectives were twofold with this study. First, to conduct a comprehensive review of state-level policies related to IV hydration spa oversight and regulation, and second, to examine IV hydration spa practices across the United States, including product offerings, product claims, and staffing. So our first study focused on the policies that govern IV hydration spas across this country. In June of 2024, we conducted internet searches using Google to find regulations and policies across the 50 states and DC. We collected regulations, policies, position statements, guidelines, and meeting minutes from boards and associations. And since IV hydration spas tend to fall under the broader umbrella of medical spas, which offer cosmetic medical procedures, they could sometimes have guidelines in states that were specifically for medical spas, but did not address IV hydration specifically. So policies that focused on cosmetic medical procedures without addressing IV therapies were excluded. The policies for each state were reviewed to determine whether policies addressed 4 categories that we determined were important for oversight. The first being governance related to regulation of administration or ownership. Second, prescriber credentials. Third, dispensing practices. And finally, compounding practices. This is a heat map showing the number of categories that were addressed by each of the states, 0 to 4. No states that were reviewed at that time had enacted legislation to regulate IV hydration spas. However, 31 states had policies that addressed at least one of the 4 aspects of oversight that were examined. 6 had policy statements that addressed governance. 12 had issued prescribing practice guidelines. 31 had regulations on dispensing, and 8 had issued guidance on compounding. Only 4 states, Alabama, North Carolina, South Carolina, and Vermont, had issued policies or statements that addressed all oversight categories. Their policies offered some variation of the following. First, that provision of IV therapy is a practice of medicine, and as a result, a licensed health professional, typically a physician, PA, or advanced nurse practitioner, must provide an individualized prescription for IV therapy. Second, compounding must generally be done in a facility that is permitted as a pharmacy by the state. And finally, dispensing of IV therapy can be performed by RNs and other licensees with medication administration in their scope of practice. Other questions regarding permitted indications for use, advertising, and on-site requirement of health professionals either varied or was absent from these states. So with this information in hand, we then moved on to our second objective, understanding spa practices across the United States. In July and August of 2024, internet searches using the term IV hydration spa and then the state were used to identify the top 5 searched sites in each state. Each site was reviewed to ascertain product offerings, product claims, and staffing, and what those were defined as can be seen here on the slide. We ultimately reviewed 255 sites in total. All sites offered IV hydration therapy, and then there were also additives in several of the sites. The additives included magnesium, NAD, and glutathione in over half of the reviewed sites. And then there were also pharmaceutical additives noted in around a quarter of reviewed sites, including ketorolac and ondansetron specifically. GLP-1 agonists and weight loss management were offered at around a quarter of the sites that were reviewed. Claims regarding the benefits of IV therapy were made by all 255 sites, but only 2 provided references for claims on their site. These claims were most often identified to be focused on immune system support through an additional analysis that's not shown here. And finally, over half of sites mentioned having licensed health professionals on staff, though fewer provided specific details about the credentials of owners or medical directors. And this took us to the final phase of the study, which was our secret shopper analysis. From August to October of 2024, 2 of the 5 sites that were reviewed in the website analysis were randomly selected and called by a single caller using a standardized script in which the caller was seeking IV therapy for cold and headache symptoms. Data was collected and coding regarding each of the questions asked by the secret shopper caller, and some of the key questions elicited by the script are shown here, including availability of licensed health professionals for questions, product offerings, claims, and any discussion of associated risks. Only 87 of the 102 sites called could answer any of the questions, and 85 answered all questions that were posed. No sites from Ohio, Rhode Island, or Wisconsin were able to answer any calls. Here are the 3 most important findings from this aspect of the study. First, of 87 sites, 24 noted that a direct consultation with a medical professional was required. The others either did not mention that they required a consultation or said that there was not a consultation required. Second, when asked about whether there was a therapy recommended for symptoms provided by the caller, 75 of 87 sites had specific therapies recommended by staff members who were not licensed to recommend or prescribe therapy. And finally, when asked about safety, only 21 of 86 sites shared general risks of IV fluids. None of the facilities also accepted health insurance coverage for the services provided, and when asked, 81 facilities provided typical payment amounts with a median of around $179 for a single treatment. There are several limitations to the study. First, we focused on publicly available state policy guidance, but can't determine really the enforcement on the day-to-day of those regulations. Second, we examined only a limited number of IV hydration spa websites and contacted even fewer for the secret shopper. So there's a possibility of overrepresentation of certain spas that might not be generalizable to all IV hydration spas. Third, we collected information through phone calls in the secret shopper study, and that can't necessarily be verified for accuracy. So it's possible there were errors in reporting or that there was potential social desirability bias. And the secret shopper questions were also limited to those that could be asked by a potential client. So we were unable to ask questions regarding compounding or prescriber practices. We also conducted our study in the summer and fall of 2024. And we can't really offer insight through the study, therefore, on practice changes since then. For instance, we did see that Rhode Island released guidance later in the summer, in July of 2024, after our policy review was conducted. And finally, there was a national shortage of GLP-1 agonist drugs during the course of our study, which may have affected product offerings by IV hydration spas. The data collected so far suggests that state regulations for IV hydration spas vary widely, and few states provide full oversight. Compounding practices in particular are minimally regulated by states with regards to IV hydration spas, which may risk patient safety. And the website data and the secret shopper analysis suggest that health claims are often made on sites with little evidence, suggesting that advertising oversight is necessary. Risks are also downplayed by those we spoke to, which may impact informed consent for these procedures. In summary, state policies vary widely, and only 4 states have released guidance that specifically addresses all 4 oversight categories that we looked at, and practices also vary widely, so more stringent oversight may be necessary to protect public health, and further studies could investigate effectiveness, safety, and compounding practices of IV hydration spa sites. Here are my references. And thank you so much to the awesome faculty mentors, Dr. Foreman and Dr. Ross, for your support, and to everyone who made this project possible. I look forward to discussing more during the Q&A. Thank you.
It was fantastic. Thank you so much. And we'll remember, we'll hold questions till the end. Next up, we have Frank Lee from Mayo on AI expert attitudes on regulation of AI tools in medicine.
Thank you so much. Hopefully you can see my screen.
Yep.
All right. Yeah, my name is Frank Lee. I'm a 3rd-year general surgery resident on research time. My PI is Dr. Cornelius Teals, a co-PI of the Surgical AI Squared lab at Mayo Clinic and a surgical oncologist. We set out to survey experts on attitudes and regulation of AI technologies in medicine. Really thank you for this opportunity. It's an honor to be part of this group. So I think it needs no introduction. AI, everywhere you turn, there seems to be some developments. And I think medicine is not immune to that. On the left is actually PubMed articles, including artificial intelligence. And then on the right is FDA submissions and approvals per year related to artificial intelligence. You see that they map pretty well, a ratio of 1 to 400. But I do think it begs the question of how do we best keep up with the speed of AI development and regulation? This was a paper in Nature a few years ago, just looking at the validation and testing of medical AI devices. They found that there were most that didn't have prospective studies, weren't assessed at multi-sites, no mention of sample size or demographics. So I think there was this open question. And I think a really big one that even we're tackling right now is how do we do LLMs, large language models like OpenEvidence, ChatGPT, and at this time that I'm aware of, there is no FDA approval for one of these such devices. And there's actually been a lot of talks. I think there's this YouTube session from an FDA session, 5 or 6 hours long, and tackling this like how do we best regulate LLMs? So our aim was, how do we do this in such a way that's data-driven, evidence-based? Our approach was to survey experts who identified as those who have published on the topic of artificial intelligence. We used their corresponding author contact email, and we sent them a 16-question survey on Qualtrics, and they ranged from knowledge questions to attitudes, future directions, and then we also offered a $5 gift card remuneration. Some examples of the questions, and thinking maybe how you would respond to these. Do you believe the FDA's current involvement in the regulation and oversight of medical AI tools is not enough, the right amount, too much regulation? Which of the following factors should be regulated for clinical AI tools? A whole host here, but just limiting it to 15 and even ranking them. So would it be accuracy higher than ethics, higher than explainability, and so on? Question 12 was who should be liable for errors made by AI? Should be the user, hospital institution, the developer, company, the FDA. And I'm not going to share all the responses just for time's sake, but we have a few preliminary results. We reached out to 629, 63 responded, so a 10% response rate. Demographics are shown here, median age 42, 61% male, most practiced in the US. And then for their roles, a lot were researcher and clinicians, a few industry and trainees, and some people who had multiple roles. In the form, we asked, what is your area of expertise? And we had drawn from, I think, a whole broad breadth of different disciplines, from medical informatics, clinical trials, anesthesiology, urology. So I think this was a really good cohort. One of the questions we asked is, what type of AI technology do you use? And I think large language models, ChatGPT, Open Evidence is far and away the biggest one. The scales for all these charts are survey responses. So more than 40 said they use large language models. Generative AI is a larger umbrella of large language models, but I think it was just very interesting to know the specificity of LLMs being the dominant tool. This was a knowledge question, true or false. The FDA has a pathway for AI-enabled devices and software for medical use and clinical applications. This is true, but about 20% did not know the answer to this. And when we looked into who they were, it's not just trainees or people who are pure clinicians. It was a whole, their demographics were varied there. This is the question about the regulation. Do we think it's enough, the right amounts? And most people actually reported not enough regulation oversight, which I found interesting. This was an attitudes question. Just, I have reservations about the use of healthcare and medical-focused AI tools for patient care currently. And most reported somewhat agree and strongly agree. And I think this maybe speaks to a healthy skepticism of what we have with this new technology. This one is about at what levels should there be regulatory oversight, testing, validation, and respondents were allowed to choose more than one. So they could have selected developer and hospital, but state had the lowest and federal was the next lowest. So that question of so many different factors, I'm just choosing the top 3 factors that were selected. But patient safety was number 1, and accuracy, interestingly, is number 2. Other things like privacy and checks and safeguards were very, very important to folks. And then the question, who should be made liable for AI errors? The user, the hospital institution, the developer were leading contenders here. And maybe it's good to hear the FDA was lowest on this one. And perhaps there's no bias in the reporting here. One of my last questions was, for obtaining FDA approval, should there be documentation submitted that discusses how bias was addressed, mitigated, such as age, sex, race, ethnicity, and socioeconomic status? And actually, this was, when we got a IRB approval, this was all before maybe recent changes as in expunging diversity in documents. But most people said they agree that this should be addressed or mentioned as to how this was addressed at least in the submission. So some early takeaways: LLMs are the most used tools. I think most report that there's not enough regulation, or they have skepticism on AI and patient care. Patient safety and accuracy were considered top regulatory factors. Liability should be shared on the developer, user, and hospital. And then bias and diversity should be explicitly addressed in the FDA submission. I think with time, I can answer some like the research lessons along the way. I thought the $5 gift card would really convince people to, just thinking, would I respond to one? And actually, most people offered not to get the gift card, which I thought was an interesting takeaway. And for next steps, we're going to do qualitative interviews. I think those questions about at what level should regulatory science be? And there's questions I didn't show, but is there a minimum level of accuracy, or what's the minimum sample size? These are all things that I hope to address in these next steps. And special thank you to the PI, Dr. Teals, and our Surgical AI Squared lab. Also to some of the coordinators like Leah Springer and Emma Benke for helping me troubleshoot the ClinCard. That was a huge role. Thank you all.
Great work. I'm sorry about the ClinCard. I know it's terrible. I'm glad that you helped though. This is fascinating, and I can't wait to ask questions about this. So next up, we have Carla Godoy from Yale talking about confirmatory evidence supporting single pivotal trial new drug approvals.
Awesome. Hi, everyone. Let me share my screen really quick. Okay, are you seeing my screen here? Perfect. Okay, so today I'm going to talk about confirmatory evidence supporting pivotal trial new drug approvals between 2015 and 2023. So I know I don't need to tell you guys what the FDA does, but to kind of set the scene, the FDA is tasked in part with demonstrating drug safety and efficacy, or rather with ensuring that pharmaceutical companies demonstrate drug safety and efficacy, which is done in large part through clinical trials. And to kind of give you an idea of what the legislation says, the law says that pharmaceutical companies must show substantial evidence of efficacy, which is taken or which can be done through adequate and well-controlled studies, which the lawyers have interpreted to mean 2 pivotal trials. And these pivotal trials are phase 3 trials. They're large studies that are designed to produce statistically significant data to support the safety and efficacy of the drug they're seeking approval for. This definition has kind of evolved over time. I think that earlier it was mentioned the FDA-MA 2.0 Act, so its precursor, FDA-MA 1.0, amended the definition of substantial evidence to mean not just 2 pivotal trials, but also 2 pivotal trials, or rather 1 pivotal trial plus confirmatory evidence. And that confirmatory evidence, as defined by also, as was mentioned earlier, through FDA guidance documents, can fall into these 7 categories, though it's not limited to these categories. And that includes clinical evidence from a related indication, mechanistic or pharmacodynamic evidence, evidence from a relevant animal model, from members of the same class, from natural history, real-world data, and expanded use of an investigational drug. So evidence can fall into those categories and more. And what we've seen over time is that since the FDA-MA in 1997, we've seen the proportion of approvals that are on the basis of a single pivotal trial go up. So immediately before the FDA-MA was approved, we saw between 95 and 97, about 20% of approvals were based on a single pivotal trial. And then more recently in 2022, we saw just over 60% approved on the basis of 1 pivotal trial. So what we wanted to do was first determine which approvals between 2015 and 2023 were approved on the basis of a single pivotal trial, then determine how many of those cited confirmatory evidence, and then characterize that confirmatory evidence, then kind of get an idea of whether confirmatory evidence was cited more frequently following a December 2019 draft guidance. So that guidance, which I've included a little kind of excerpt here, clarified what could be used as confirmatory evidence, what confirmatory evidence was, and was meant to really give pharmaceutical companies a guideline for how to treat confirmatory evidence. So we wanted to see if that guidance led to more frequent citation of that evidence. So how we did that, we identified the approvals between those years using the FDA Novel Drug annual reports, which is essentially just a list, or is a whole document that's published each year, which includes in part a list of the drugs approved that year, and then kind of outlines a few different characteristics. So what special pathways were used to approve the drug, things like that. And then I can show you here, these are the different sort of pathways that can be used to approve, or accelerated pathways to approve drugs. And these all provide regulatory flexibility for drugs that either treat diseases that are very serious, maybe that have no other treatments, or that can offer that are better than existing treatments or than the standard of care. We examined, in order to find the confirmatory evidence, we actually looked through the FDA's review documents, which are available publicly online, and actually went through and read the different documents, and then classified that confirmatory evidence according to the 7 categories that I mentioned earlier. We included evidence that we deemed that was explicitly mentioned, so using the words confirmatory evidence or very similar language, or we also included implicitly designated confirmatory evidence. So phrases that could be easily construed as indicating confirmatory evidence. And this is kind of an example of what one of the review documents looks like. And so here you can see that it mentions confirmatory evidence. So this would have been one that we classified as being explicitly designated as confirmatory evidence. We excluded a sample of, or a portion of drugs that were single pivotal trials approved with at least 1 clinical primary efficacy endpoint without orphan drug designation. So we actually pulled the data from this cohort of this population of drugs and found that they all had very large patient populations, were across several countries, across several centers. And we found that this kind of fit the FDA's definition of what could be considered sufficient for demonstrating substantial evidence of efficacy based on one trial alone. So this population was excluded. And then for analysis, we use descriptive statistics to characterize a sample, and then chi-square test to compare before and after that 2019 draft guidance if confirmatory evidence citation changed. And then what we found, as you can see here, was that about 45% of the approvals between those years were on the basis of 2 or more pivotal trials. And then about 8.9% were in that excluded category. And then 46% were approved on the basis of a single pivotal trial. So this doesn't account for whether it was just a single pivotal trial or if it included confirmatory evidence. And then the next graph will kind of break down this leftmost slice of the pie, which you can see here. So we have about 20% that explicitly reference confirmatory evidence, 77% that did not reference any confirmatory evidence, or we have the leftover slice right here in which confirmatory evidence was implicitly referenced, which was about 4%. And then you can see here kind of macroscopically at a glance that over time, this darker blue represents the approvals with confirmatory evidence. You can kind of see that this has gone up over time, but to break it into actual numbers, of the 40 approvals that referenced confirmatory evidence, about 7.6% were between 2015 and 2019, whereas 34% or almost 35% were between 2020 and 2023, we found to be statistically significant. And then to give you some more statistics, about 40 approvals referenced 97 unique sources of confirmatory evidence, which tells us that some of these approvals referenced multiple sources. The most common types were pharmacodynamic and mechanistic evidence, which was 49, and other. Of the other, the most common were results from a non-pivotal trial. So this was just any additional trial that was included that was not the sort of larger pivotal trial or the main pivotal trial, supporting the results of that main trial. And then we didn't find any instances of real-world evidence, clinical evidence from a related indication, and evidence from members of the same pharmacologic class. And then about 86% had at least one special regulatory pathway designation. So those are the ones that I mentioned earlier. So fast track, breakthrough, all those different ones. And we found that breakthrough, priority review, and fast track were the 3 most common. So the limitations, unfortunately, we can't read minds. So that made it difficult to know or could limit sort of because of the variability in language that the FDA uses, limited whether we were able to fully kind of understand when they were maybe implicitly referencing confirmatory evidence. And so we only were able to categorize when explicit or implicit terminology was used. And then we also were conservative in our simple eligibility criteria by excluding, like I mentioned, that population of drugs that had at least one clinical primary efficacy endpoint without orphan designation. So to kind of recap, we're seeing a rising number of FDA approvals on the basis of a single pivotal trial, and also increasing reference to confirmatory evidence. Also, a large proportion of that evidence doesn't fall under those 7 predetermined categories outlined by the FDA. We also saw a surprising lack of approvals that reference real-world evidence or clinical evidence from a related indication. This could be in part just because it's hard to collect real-world evidence for medications that haven't yet been approved by the FDA. So this could play sort of a larger role in supplementary new indication approvals, which is not something that we looked at for this study. And then overall, our results may signal that it could be useful to gain greater clarity and specificity on the use or on confirmatory evidence standards and communication of its use. So further clarifying for pharmaceutical companies what exactly the FDA would like to see in terms of confirmatory evidence. And then I'll take questions at the end, as we said. I wanted to say thank you to the whole CRIT team and to Dr. Ross, who helped me a ton with outlining this project. Thank you.
It was great work. Thank you so much for presenting it today. And last but not least on this panel, we have Garrett Regan from Mayo Clinic discussing co-pilot diagnostic tools for clinical trials of anti-fibrotic Crohn's disease therapeutics.
Yeah, thank you. Are you guys able to see my screen all right?
Yes, looks great.
Yeah, so as mentioned, my name's Garrett Regan. I'm a PhD candidate at Mayo Clinic in Dr. David Holmes's lab, and I'm excited to talk to you today about my project around AI-based tools for clinical trials of Crohn's disease therapeutics. So just as general overview, I'll talk a little bit about the background of Crohn's disease, and specifically the problem that we face with intestinal stricture localization, the solution that we've provided through some uncertainty-aware segmentation techniques, the results that we have of a few experiments, and some conclusions that we've drawn from our data. So Crohn's disease is a chronic inflammatory disease of the gastrointestinal tract that affects about 1 million Americans. And it's a complex condition that frequently presents with these stricturing or penetrating complications. And I'll more specifically be focusing on the stricturing subtype for this presentation. But an estimated 50% of patients will develop these complications within 10 years of their diagnosis, posing a massive risk for hospitalizations and further complications down the line. So an intestinal stricture is just a narrowing of the intestinal lumen caused by a thickened intestinal wall brought on by complex remodeling of the intestinal wall due to the presence of chronic inflammation characteristic of the disease. And strictures most commonly present in the terminal ileum of the small bowel, and as I mentioned, are a leading cause of morbidity amongst Crohn's disease patients. I'm sure many of you have seen commercials for things like Humira on TV. These are biologic drugs that are targeted at decreasing the inflammatory presence of Crohn's disease. And though there's many drugs out there, many of them do only target inflammation. And while these drugs can help induce and maintain symptom remission, they often have little effect in treating existing fibrotic stricturing disease. So as a result of that, about 80% of Crohn's patients are going to require some form of surgery during their lifetime, the most common indicator for which being intestinal strictures. And surgery is not without its own problems as well. Though surgery can sort of eliminate those diseased bowel segments for a time, they often recur in about 50% of patients. So due to those factors, there's been interest in recent years of trying to use anti-fibrotic drugs for the treatment of intestinal strictures. And these have been pretty promising so far. There's currently a few phase 2 clinical trials recruiting for evaluating the safety of these existing anti-fibrotic drugs in people with fibrostenotic Crohn's disease. But the problem that we still face as these early anti-fibrotic drugs are sort of on the horizon is that there's several issues that still need to be addressed as far as how we go about diagnosing and tracking strictures across patients. So these two main issues are strictural localization and stricture measurement, and I'll predominantly be focusing on stricture localization today. So the location that a stricture affects in the intestines is just one piece of a very complex reading paradigm that radiologists must follow when they're trying to diagnose and track these strictures, both in clinical trials and in clinical practice. So the idea that we have here is, can we in some way automate the stricture localization piece to try to reduce the burden placed on the radiologists? Functionally, using deep learning techniques, localization can be achieved through standard image segmentation. But in healthcare settings, we rely on sort of these non-technical experts to interact with these AI models, but they also, in a sense, need to be able to trust the predictions that these networks are producing. And that's where the notion of this sort of uncertainty-aware deep learning techniques come into play. So there's a lot of different methods out there for quantifying segmentation uncertainty. A few listed here are deep ensembles, Bayesian neural networks, Monte Carlo dropout, and test time augmentation. And this first group here addresses the quantification of epistemic uncertainty, which is the uncertainty in your model parameters given a lack of training data. This last one here, test time augmentation, addresses aleatoric uncertainty, which is irreducible uncertainty that actually arises from the data itself, which is what we'll focus on today. So augmentation is a very common technique that's used across many deep learning pipelines to sort of artificially increase the diversity of your training data when you're training a model. When you then apply augmentation during test time, though, you generate an ensemble of predictions that you can aggregate results from, generating a consensus segmentation that is more robust to these image transformations that you've applied. But you can also generate a predictive entropy map that highlights these areas in an image that may be more uncertain than other tissues in the background. So today, most segmentation networks are generally built on a standard UNet architecture, and many of them use the NN-UNet framework, which is just a self kind of configuring UNet architecture that adapts to its training data. And sort of conveniently for us, all of the NN-UNet implementations out there share the same family of augmentations, meaning that one test time augmentation pipeline that works for one implementation should theoretically work for all of them. So as a proof of concept for this study, we implemented a test time augmentation pipeline for Total Segmentator, which is a publicly available segmentation network built on NN-UNet that's capable of segmenting over 100 anatomic classes. And we evaluated it under the task of multi-organ abdominal segmentation. We evaluated this study on 872 CT cases with known pathology, which I'll get into a little bit later. So we were able to generate organ and augmentation level uncertainty estimates by aggregating computing the average entropy within dilated segmentation mass for each one of our organs of interest. And some observations here, the intestines, which will be the focus later down the line when we get focusing on strictures, were some of the most highly uncertain organ classes that we examined, just showing the difficulty that we might face down the road. But on the augmentation side, image rotations produce a slightly higher but significantly higher bump in the median uncertainty across each one of these anatomic classes that you can see in the heat map below. We also noted that uncertainty seemed to peak in areas of known pathology. So we had ground truth segmentations for tumors of the pancreas, liver, and colon. And each of these three cases on the right show areas where the average uncertainty within the segmented tumor was higher than that of the background organ itself. So the question here became, can we use this test time augmentation pipeline as sort of a generalized early stage detection tool for pathology? So we did a study here where we compared the average uncertainty within cancerous tissue and healthy tissue. And in each one of those cases across the pancreas, liver, and colon, the cancerous tissue yielded higher uncertainty than the healthy tissue. And we also conducted a receiver operating characteristic analysis at 1,000 different uncertainty thresholds, try to identify how well the uncertainty information itself can be used at detecting pathologic tissue. So some of the results from this you can see in the pancreas and colon, we had sort of moderate success at detecting these pathologic areas, AUC values hovering around 0.7, but we actually had fairly high success at detecting liver tumors with an AUC value of about 0.9. And this is largely probably due to the fact that the average uncertainty in the liver was comparatively much lower than the pancreas or colon. It's important to note, though, that in this study that even the false positive results that we generate are still indicative of uncertain segmented regions that in an active learning pipeline or some kind of iterative segmentation technique might still need to be updated manually by a user. So they still provide value to the user, showing where there's truly uncertain segmentations along with potential pathology. So just in conclusion, I presented today a method that we developed for quantifying segmentation uncertainty and pathology detection using publicly available segmentation networks. Some next steps will be to compare how this performs across more disease tasks outside of the pancreas, liver, and colon. And then we need to begin to adapt this technique for MRI to focus more specifically on intestinal segmentation and specifically intestinal stricture detection. So some implications then for regulatory science. This has the potential to improve the efficiency of that reading paradigm, which I mentioned previously for radiologists in clinical trials and clinical practice, hopefully allowing us to better understand and better identify novel Crohn's disease therapeutics in the future. So that's all I have. Thank you, everybody, for listening. I'll be happy to take any questions shortly.
Great work. Thank you so much. So with that, we have about 13 minutes left for questions. Does anybody have any questions they want to start us out with? So there's a question in the chat. What percentage of the IV clinics had any type of accreditation?
That's an interesting question. When we were looking at or collecting data, we specifically looked for credentialing of the staff members in the clinic. So when we looked for licensure information, but a lot of websites actually didn't provide that information on their sites. So it was difficult to actually assess that. Accreditation, in terms of what that accreditation would be, that's also a good question, since there's no real standard for these facilities at this time. So I think moving forward, that's something to really consider. How are we actually, what standard do we want to hold facilities that are providing IV therapy to?
How old are these? Like, how long have they been around?
So it's actually also an interesting question because there's not a lot of literature out there, but we looked at Google search trends, and starting around 2010 was when the term IV hydration spa began to really blossom. So it's a really fast growing field, and the market research also shows that the field is growing by billions every year. So it's pretty significant.
That's fascinating.
Do you envision this, I guess, as something that the FDA will step in and actually regulate, or do you think it will be more so under the sort of umbrella of like supplements that are really sort of blind to the FDA as far as regulations go?
I think at this point, it would be a really big change for the FDA to step in and regulate these facilities because they are considered compounding pharmacies, and that is something that's typically left to the states for regulation if they are not registered as voluntary outsourcing facilities. But there might be a paradigm shift that comes in where people might, or facilities might want to register as outsourcing facilities to, reflect the quality of their service. So that's something also that might occur, like to improve, to allow for federal oversight. We'll see.
Something I thought was really interesting when Frank gave his presentation is that when you asked the AI experts what kind of regulatory regime they preferred between federal and state, they preferred federal, but it's exactly the opposite with what Anishaa has. Does either of you have any kind of comments on that, state versus federal?
I don't. I think it's. I think that's probably a good question for when we do our interviews. There's more consistency at the federal level that people can think of, especially for such a broad technology that really does not, it's not really tied to geographic boundaries.
Mm-hmm. Something I was actually really surprised by with your findings, Frank, is how, I mean, not dubious exactly, but sort of like how cautious your AI experts were about the quality of AI, which really surprised me because this is what they do, right? And this is what you do. So I would love to hear your take on kind of how would you have responded to your questions?
Yeah, I think when I had my own suspicions for some of them, so one of them, well, two questions I didn't show was either or, do you believe there should be a minimum AUC or a performance metric for all these models, or as long as you report it in your submission in the summary statement that's acceptable? And actually I thought people would be, as long as people are aware, then they can use it knowing the limitations. But actually most people said that they do want a minimum accuracy threshold. I didn't have the follow-up question of what that would be. The second one was, I thought, is there a minimum number of data points or sample size? And I thought they would kind of the same thing. It would be so context dependent, but so many people were willing to say there should be a minimum data point size. And they even gave numbers anywhere from 300 to 10,000. So I think that's just, I don't know, I think there's a lot of nuance, and hopefully the interviews will help.
It was very impressive, and maybe a statement on how interesting your survey was that so many people were willing to be interviewed. So I'm really curious to hear kind of what your follow-up is there.
Yeah.
Other questions?
Just to follow up on that, I was curious for Frank, what I know that a lot of them were concerned about patient safety. I was curious if you delved into that more in detail about what aspects they were worried were going to impact patient safety.
No,
Was it the
but
accuracy or okay?
Yeah, no, I think those were just kind of when we were designing this. We wanted to be approachable for all comers, and I think the patient safety question will differ between if you're industry or a researcher, clinician, and I think ultimately that's an easy, like a kind of a lightning rod for something that I think is a clear line in the sand. Like I think things like patient safety, privacy are things that we clearly understand that should be regulated. Whereas I think a lot of the other things, maybe the 15 things are nice to have, but people would argue might not necessarily need to be regulated. No, that's a good question. And then Jeffrey, your point of so many people being interested in being interviewed, I think that like I had a line there for the bias on who would want to fill out a survey. But I think the people who are really interested, like there was an oopsie where I didn't publish the most latest version of my survey to pay people back for the $5 gift card. So I actually had to email a few people and just say, hey, do you want this? And then from those emails, the results were still all anonymous, but I just knew that they had filled it out. From those emails, a lot of people wanted to be like, you know, I work in this field, or I'm leading a task force or strategic group at my institution on AI regulation. And I think that's where the interest in wanting to talk about it is. You have people who are, they enjoy doing this. They don't need a $5 gift card to convince them to fill out this survey.
Yeah. Carla, this is Joe. It's funny, as you were presenting your project, I was thinking a lot about the presentation from Dr. Kozlowski, who talked about the sort of the challenge of developing new methods, essentially, and like the sort of how that has to happen and the trust within the industry to then for them to submit that data. And I'm wondering just what, if you could reflect on it in terms of this concept of confirmatory evidence. And, you know, there was a big uptake after the guidance document, but obviously not every company provided quote unquote confirmatory evidence. Like, I'm wondering, because I know you spent a lot of time reading through all those approval documents, if you got a sense of whether this was evidence that had been discussed with FDA, and so the industry was highlighting it, or was it being put in as extra in the hopes of sort of advancing what the way the agency works and the way they handle confirmatory evidence?
So you're saying the evidence included by the pharmaceutical companies, if that was added as like an additional bit of evidence, or if it's something that was already included?
Right. Did they acknowledge sort of even explicitly, like, well, we saw FDA's guidance, so, you know, here's this, you know, like, it's just about the sort of the process of change and how it happens.
Yeah, there were a couple that did actually cite the guidance document and said, like, because of this guidance document, and they actually copy pasted parts of it and said, because of that, we will include this evidence, or we have designated this as confirmatory evidence. There were a few instances, there weren't very many, but certainly, like, I think that the clarification from the 2019 draft guidance did make a huge difference, because I think previously it wasn't that companies didn't want to include the evidence, because a lot of the evidence was existing evidence anyway that was collected just as part of the development process. I think once the FDA was able to put language to it and clarify what they wanted, pharmaceutical companies were better able to identify it within their NDAs. So that's kind of my answer. I'm glad that you said this is Joe. I haven't seen you in months, so I wouldn't recognize you otherwise.
He's been marathoning. I actually have kind of a similar question to Garrett, which is I was thinking about Dr. Kozlowski's presentation while you were giving your talk, because I mean, you're kind of doing what he was talking about, right? You're doing the development of some of these things that could be used in regulatory processes in the future. And kind of, how do you think about your the place of your technology that you're developing within that, or is that something that is just kind of off your radar?
No, yeah, I think it's really important to try to, as he mentioned, try to evolve technology as the regulations evolve as well. And I think a big piece of what is going to come into play here is how you analyze uncertainty with these tools across clinicians, right? So whether or not you can actually trust the predictions that are being generated by a tool is informative, right? Rather than just generating a prediction, generating how confident is that prediction on top of it is going to be very important in trying to identify how well a tool was able to generalize and how well a tool was able to be extrapolated into different scenarios.
Thanks. Any last questions? Well, this was just fantastic work by all of you. I'm so glad that we had the opportunity to hear today and that we had the opportunity to provide you a little support to further your research. Congratulations to all of you.
All right. Thanks to those who just presented. Let's move to our second session. And the first speaker is Alyssa Columbus from Johns Hopkins University, CERSI, talking about strategies to assess and report variability arising from multiple analytic choices in biomedical data analyses. Thank you, Alyssa. Take it away.
Thank you. All right. Hi, everyone. My name is Alyssa Columbus, and I'm a third-year biostatistics PhD candidate, inaugural Vivian Thomas Scholar, and CERSI Scholar at the Johns Hopkins Bloomberg School of Public Health. And I'm thrilled to be here today, and I'm excited to go over my research, which looks at both a bird's eye view and on the ground at how we can both qualitatively and quantitatively improve the rigor, reproducibility, and transparency of biomedical data analyses. In other words, today we're going to dive into strategies for analyzing the process of data analysis and making it more trustworthy by noting and reducing unwanted variability, which is absolutely critical to having robust and reliable regulatory decision making. Before I get into my work, though, I just want to start with this powerful quote that has inspired much of my work, which was said by Dr. Alondra Nelson, the former acting director of the White House Office of Science and Technology Policy, at the 2022 G7 Science Ministerial Meeting. And her quote was this: "Openness and security are not contradictory, they are complementary and mutually reinforcing." In other words, when data and the means of generating evidence are made public and open while preserving people's privacy, it helps others verify that our conclusions are accurate and honest and can be trusted at the same time while safeguarding people's well-being. And translated to an even broader scope, this delicate balance fundamentally drives modern medicine, public health, and the effectiveness of regulatory decisions that affect millions. From evaluating the efficacy of new medications or treatments, to shaping clinical guidelines, to navigating exactly what to do and when during a pandemic, regulatory bodies like the FDA rely heavily on consistent and transparent data analyses. But here's the catch with all of that. Even seemingly small analytical decisions during the course of a larger analytical project, like how a researcher may manage missing data or choose what variables to include in their analyses or in their models, or how they apply specific statistical methods as opposed to others in their team, can drastically influence the outcomes of their research. And depending on how each of these is communicated, public trust and regulatory credibility can be affected as well. So how can we manage these immensely impactful yet frequently hidden choices? Well, that's a big question. So let's start small by looking at the process of data analysis itself and how it, step by step, works to advance our knowledge and understanding. Standard models of data analysis, like the one shown on this slide, often depict the process of deriving understanding from data as an overly structured and not very fluid process at all that ends at some ambiguous unknown point. But as many of us have experienced firsthand, real-world data analysis is anything but structured or final. It's iterative, and it's always changing, and it's often shaped by some combination of ethical guidelines, stakeholder demands, organizational realities that we have to deal with, and high stakes of legal and regulatory scrutiny. And recognizing and explicitly addressing this complexity is very important, not just for theoretically and mathematically describing the process of data analysis, as these authors have done, but also for translating it into actionable findings and even guidelines to make better, more effective data analyses. So that's where my research comes in. I recently led the first ever research study called The End is Insight that asked the question, when is a data analysis done? And investigated how different scientists across fields know when their process of understanding is complete. And I interviewed these scientists with 10 to 50 plus years of experience in many subsectors of academia, government, and industry. And regardless of how long they'd been working or where they'd work or what they've done, they all highlighted how balancing methodological rigor with real-world constraints like resource limitations and stakeholder pressures is a continuous challenge, and they also consistently emphasize the importance and difficulty of transparently documenting every analytical decision that they made. And from all of their insights, I created this practical visual light bulb shaped roadmap on this slide, which clearly lays out the essential phases of analysis in a fluid way, along with iterative feedback loops and explicit transparency checkpoints. It's basically a light bulb shaped roadmap to insight and a complete data analysis. And all of this was crafted in such a way that it can be tailored to align with any needs and expectations, including those that have more stringent regulatory requirements or require more oversight. Building on this work, instead of continuing to look to the conclusions of data analyses, I'm looking at the conclusions of different data analyses. And it has been demonstrated many times in the literature that even though a data analysis may be done, it doesn't mean that it's reproducible or accurate, even when using the same exact data set. And repeated efforts can sometimes yield entirely different, in other words, a multiplicity of results, like the ones I show in this branching tree here on this slide. And even repeated independent analyses of the same data can inadvertently skew findings by reducing the heterogeneity of results, confirmation bias, and inflating false positives, which can be a serious problem for agencies like the FDA that depend on replicable, accurate, and unbiased results. Now, simulation studies can clearly demonstrate these risks, and we've identified practical strategies to address them, including the rigorous pre-registration of analytical plans, auditing and validating these plans and results using both the original and additional independent data sets, and frameworks like the vibration of effects framework, which can quantify how different decisions made during the analytical process can serve to reduce or to increase the severity of results uncertainty. Now, this is all continuing work, and I plan to further quantify these effects and draft guidelines to help researchers everywhere build more secure and trustworthy data analyses that ultimately yield more robust research outcomes. Now, all of the work I've done so far hasn't just been mathematical or theoretical in scope. It has already directly shaped real-world policy and practices, and I've been involved in NASA's open source and open science initiatives for almost a decade, and my efforts have drawn significant attention and recognition from the Science Mission Directorate, the Ames Life Sciences Data Archive, and the Gene Lab. And I've also published a policy memo that develops specific actionable recommendations for transparency and reproducibility with patient privacy in mind, including clear data sharing protocols and researcher training, which has received attention from bipartisan lawmakers since it was published last year. Also, beyond the United States, my research papers and policy recommendations have been cited in over 60 international policy documents written by major global organizations like the World Health Organization, the Organization for Economic Cooperation and Development, and the Council of the European Union. And it's further been cited by other regulatory agencies and governments globally, as reflected on this map, who all want and need to be able to develop robust and trustworthy international health policies based on the highest quality of evidence. Of course, none of this work has been or is currently being done alone. I'm deeply grateful to my co-advisors, committee members, and the many other international mentors, colleagues, and organizations that have supported me, from Johns Hopkins to Stanford and beyond. Their guidance and insights have been instrumental in turning my statistical work into meaningful policy and practice. So in summary, enhancing transparency and reproducibility in biomedical data analysis isn't just beneficial, it's essential. And this ethos and all of my work that supports it is about far more than better science. It's about building stronger regulatory and policy frameworks, and as a result, more evidence-based and trustworthy public health decisions that we can all benefit from. So thank you all for your attention, and I'd like to give a special thank you to my advisor, Brian Caffo, for being here today and for all his support, and to Michelle and the other event organizers for putting all of this together. And I'm excited to hear your thoughts and answer your questions during the moderated Q&A. And if you have any questions at any time, including after today's seminar is over, please feel free to email me or contact me via my website, which is just my first and last name, melissacolumbus.com. Thank you.
Thanks, Alyssa. That was great. You're really raising the bar on CERSI Scholar presentations by putting a map of the world up and showing everywhere where your work has had an impact. Our next presentation is from Shifang Liu from Yale, who's going to talk about new while alive regression models for analyzing composite survival endpoints.
Okay, thank you. Can you guys see my slides?
Yes, we can. Thank you.
Okay, great. Thank you all. My name is Sifang. I'm the postdoc from Yale, and the project I'm going to share today is about a wildlife regression analysis of composite survival endpoints. So from this title, we can see this project involves a composite endpoint and a regression and in clinical trial. So to begin, first, what is the composite endpoint and why it is important? A composite endpoint actually combines multiple individual outcomes into a single measure. For example, in a clinical trial, each participant may experience multiple types of events, for example, hospitalization, non-fatal MI, and the event of death. In composite endpoint, instead of analyzing each of these events separately, we combine them into one outcomes. One outcome, we can either treat each events equally or assign different weights based on how important each event are. And we call this weighted sum. Another way we can do so is we can simply focus on the time to first event. One of the main benefits of this composite endpoint is it actually can significantly reduce the sample size requirement, and this is a big deal for the study design. The idea of composite endpoint is new. It's actually several regulatory guidelines have emphasized this, like SHE9 and some FDA guidelines have emphasized that not only how important the composite endpoints are, but they also support to use this composite endpoint as an alternative to the traditional endpoint. They also highlight that we need a clear definition and some proper analysis when used the composite endpoint. So in this project, we want to take advantage of this composite endpoints concept and to build a regression model for the time to event data. And our goal is to estimate the time varying effects. And this approach can help guide the personalized medicine by relating the individual risk factors to the cumulative events change over time, and it also can better reflect the complexity of the real-world clinical event, a clinical trial like the oncology trial. A good example to motivate our work is the HF action trial, which we are interested in to look at how exercise training impacts a composite of all-cause death or all-cause hospitalization. So in this trial, there are definitely two types of events, the hospitalization and deaths. However, if we only focus on the time to first event, we may miss all the subsequent information like repeated hospitalization, and this can lead to biased conclusion or the misinterpretation. There are several methods has been proposed to analyze such complex data, which includes the terminal events and several types of recurrent event. For example, there are methods for recurrent event that treat the death as sensory or some cumulative frequency approach. However, all of these well-developed methods ignore something really important. The fact is, if a patient dies, their chance of experiencing other events drop to zero. So for survival times actually plays a huge role in how often we can observe these events. And the cumulative frequency approach alone doesn't account for that. That's where the while alive idea comes in. It's designed to capture both how often the events happen and how long the patient is actually alive to experiencing them. So what is the while alive and how it works? Formally, the while alive loss rate is the expected number of the weighted events. It actually includes both recurrent events and fatal events, and they are summarized with different weights based on their importance. And it also divided by the expected time the patient is alive. So in the denominator part is actually the restricted mean survival time. What we are doing here is actually we calculate the average cumulative loss per unit of survival time. You can think of it as a survival adjusted loss rate or a survival completed version of cumulative frequency. Let me give you a quick example about the weighted sum cumulative frequency. For example, imagine we have 3 types of events, with 2 of them are recurrent and 1 of them are fatal. If we decide that the death counts as twice as important as recurrent event, then even for the first two patients, they have different event history. Their weighted event counts end up the same. This actually allows us to compare the patient events fairly, because in most clinical trials, we consider the death as the more important, more severe event compared with other non-fatal events. So in our project, we're building a regression model for this while alive loss rate, which allows us to model time-varying covariate effect. And this is important because the concept of wildlife is actually time-sensitive. It changes over time. And it also depends on how long the patient survives. So it makes sense that the effect of the risk factor should also vary over time. If we just ignore them and consider as fixed, we are missing part of the picture. Here we allow a flexible link functions based on our research questions and our interest, and we can use an estimating equation to estimate the model parameters. However, in practice, we run into a challenge that we don't always observe all the events due to censoring, like when patient drop out or the study ends. So to handle that, we propose a weighted generalized estimating equation methods. And beside that, we use the spline to stack the data and model the time-varying effect and just for the censoring properly. We have done some simulation studies to test these methods, and the results looks pretty good. Our estimator is unbiased, and the variance estimates are accurate, and the confidence interval coverage have a coverage probability close to the nominal 95%. Later on, we apply these proposed methods to our initial motivating example, the HF action trials. Here we adjust for various type of the covariate effect, and our goal is to estimate the time-varying intervention effect and the covariate effect as well. Here I show several the estimated effect trajectory. You can see some of the covariate effect change quite a bit over time, and also some of them has a time varying statistical significance. They shift across the follow-up period. This kind of insight would be hard to get if we only simply analyze the data using some traditional methods. Looking ahead, we plan to extend these methods in a few directions. First, we want to handle the cluster randomized trials, where the participants are grouped into clusters and their outcomes are correlated. We will study how the methods performs and their asymptotic properties in these settings. Second, we want to develop a data adaptive way to choose the number of spline knots, because we use a spline to model time varying effect, and we usually need to decide how many knots we need before that. So this helps us to provide a more flexible model for the time varying effects. Finally, we plan to build a user-friendly R package so the researchers can easily apply our proposed method to their own research data. Thank you all. This is Anna, and I'm happy to take any questions at the end.
Wonderful. Thank you, Shifang. This is just to show you how broad regulatory science can be. So I am not a biostatistician, so I'm going to make sure that everyone knows they're able to ask questions at the end, because people will have more sophisticated questions for you than I might. But we'll go to our next speaker, Xiaozhou Zhou, who's going to be talking about implementing the principal stratum strategy for intercurrent events in the CALGB, C-A-L-G-B 90206 trial, a case study of an oncology randomized trial. And Xiaozhou is from Duke, the Triangle CERSI.
Hi, thank you for introduction. And can everyone see my screen now?
Yes, we can.
Oh, yeah, great. And yeah, my name is Xiaozhou Zhou. I was a postdoc at Duke University, and now I was a faculty at UAB. And today my presentation project is implementing the principal term strategy for intercurrent events in the CAG-LGB 906 90206 trial. So it is a case study in oncology randomized trial. I'll start with the introduction of this trial, and then I will describe the principal strategy briefly, and proceed to result interpretation, and then I'll list some future work. So this trial is a phase 3 randomized controlled trial in metastatic renal cell carcinoma, and it recruited 732 previously untreated RCC patients between 2003 and 2005. Half of them were assigned to a treatment arm, namely the bever plus IFN arm, and nearly half of them were assigned to the control arm, namely the IFN monotherapy arm. So the primary endpoint of the trial is overall survival. The original intention to treat analysis showed a non-significant trend favoring the bever plus IFN with a p-value of almost 0.1. However, the high rates of treatment discontinuation arising from various reasons, such as toxicity, disease progression, were reported. So it means that the original intention to treat analysis only reflects the effectiveness of the treatment, not the efficacy. So our aim is to assess the efficacy by focusing on the subgroup who would continue the assigned treatment and both assignments. In fact, a treatment discontinuation is one typical intercurrent event by the SHE9. And the formal definition of SAE is the post-randomization events that can affect either the interpretation of the existence of measurements relevant to the clinical question of interest. And the SHE9 also advocates the estimate framework to define the treatment effects of interest accurately. So it specifies 5 strategies to handle the SAE. And thank you for thanks to she for introducing some of the strategies. I think for her project, she focused on the composite strategy with a composite endpoint. And for us, we focus on the principal stratum strategy. The key of principal stratum strategy is the principal structure. You can understand it as a label of each participant, based on which we can classify each participant to the subgroups. So it is defined by the joint potential values of an intercurrent event variable and both treatments, the D1 and control, the D0. With a binary discontinuation, so we can have the full structure here. And the U equals to 0, represents the participants who would continue the assigned treatment regardless of their initial assignment. And the U equal to 1 means that the participants who would discontinue the treatment if assigned to the control arm, but would continue if assigned to the active treatment arm. So for the remaining 2 structures, they can interpret it in a similar way. And the key idea of this strategy is that the stratum membership is conceptually defined before randomization, so it allows for the causal comparisons within strata. However, the challenge here is that the structure is latent because we can only observe one intercurrent event for the assigned treatment. It means that if the patient is assigned to the treatment group, we don't know if he will discontinue or not and the control arm, right? So for the estimation, there are generally 2 approaches. One is a mixture modeling approach with these 4 assumptions, and 2 of them are optional. And the other one is a weighting approach with a principal ignorability assumption. So we also developed the R package called the pStructure, and to handle various intercurrent events and various outcome types. Here is the QR code to our manuscript. Feel free to scan and check out our package. And then we reframe our question into an estimate framework. We specify the 5 attributes within this framework: the treatment, the population. So for the population, we focus on the patients within the target principal strata, namely those who would never continue assigned treatment regardless of their initial treatment. And the endpoint is overall survival. The intercurrent event is the treatment discontinuation, and the population level summary is the difference in survival probability over time between 2 arms, specifically within this target principal strata. Here is the baseline characteristics by the arm and the discontinuation status in the trial. We observed that the difference in this kidney removal history and having more than 2 risk factors, we can say the rate of these variables are higher in the discontinued group than the continued group, regardless of the treatment arm. So here we present the survival probability and each treatment across the 4 strata. And this here is a causal effect on the survival probability across 4 strata. So we can say the potentially beneficial effect of the bevo plus IFN in this and this strata. However, for this strata, which is our focus, the effect is minimal. And for this strata, it showed a potentially harmful effect. So the treatment effect is heterogeneous across 4 strata. And here we present, here is the estimated proportion of 4 strata. For the never discontinued strata, which is our focus, the proportion is about 44%. But a caveat is that the results showed large uncertainty reflected by the wide confidence bands, especially for this smaller strata. So our future work is to complete the sensitivity analysis in both methods, since we involve some assumptions here, and we will also conduct more simulation studies to evaluate the performance across a wide range of settings. And also, since the principal strategy is based on the latent principal strata, so we will create improved visualization methods and diagnostic tools for understanding this latent strata structure. Here are some reference, and we thank the Oncology Center in FDA for the support, and also we thank the Triangle CERSI for the guidance. I'll take questions after the talks, and thank you.
Thanks, Xiaozhou. That was great. Very, very interesting. Okay, we have one more presentation in this session from Xiaoqi Tong at Yale. The title of the talk is Partial Principal Ignorability Assumption with a Novel Odds Ratio Sensitivity Parameter to Point Identify PCEs. So thank you, Xiaoqi.
Hi, everybody. Can you see my slides?
Yes, we can.
Good afternoon, everyone. My name is Jiaqi Tong. I'm a second-year PhD student in biostatistics. Today, I'm going to present my research project on addressing intercurrent events in causal inference. Intercurrent events are post-treatment events that may affect either the interpretation or existence of final outcome, which is very common in clinical trials and observational studies. principal stratification is a popular causal inference tool that can address the statistical challenges in the presence of intercurrent events, which is recommended in guidelines in clinical trials. Under principal stratification, one treats the joint potential values of the intermediate outcomes as pre-treatment covariates. Based on that, one can classify the whole populations into 4 principal strata. For example, under truncation by death, quality of life is not well defined for individuals who die. A direct comparison among survivors of quality of life outcome may lead to selection bias. In this case, one can rather define a scientifically meaningful estimate among subpopulations who would have survived independent of treatment assignment, which is so-called survival average causal effect. The second example is non-compliance, where the treatment received does not align with the initial assignment. And similarly, in this case, we can also define a relevant estimate among those who would have always complied to their treatment assignment. We assume that an analyst can access to a data factor consisting of a final outcome Y and a binary intermediate outcome D and a binary treatment assignment Z and a vector of baseline characteristics. And we will pursue the potential outcome framework in causal inference and define YZ and DZ as the counterfactual final outcome and the counterfactual binary intermediate outcome. This table illustrates the joint potential value of intermediate outcome under non-compliance. We can see that when D represents the treatment received, we can divide the whole populations into 4 principal strata, which are always takers, never takers, compliers, and defiers. Because the principal strata variable D0 and D1, the vector is latent and is not fully observable. We typically rely on some additional structure assumptions for point identification. In the existing literature, there are 2 popular competing approaches. First, monotonicity, which under truncation by death assumes that the treatment is not harmful. And second, the counterfactual intermediate independence assumes the cross-word conditional independence. However, both approaches are sometimes criticized for their limited applicability in some scenarios. For example, the monotonicity is very difficult to justify when comparing to active treatments, and the counterfactual intermediate independence may be also overly strong in some scenarios. In this project, we will propose a novel sensitivity analysis framework to relax these assumptions. Comparing to the existing sensitivity analysis framework, our parameterization by odds ratio has 3 key advantages. First, the odds ratio is more familiar and interpretable for practitioners. Second, the odds ratio parameterization is statistically better than the existing approach. For example, the existing approach uses the ratio of strata proportions as their parameter, but their parameter range is constrained by some unknown marginal distributions, which may cause operational challenge. In contrast, our odds ratio parameter is margin-free and variational independent of the marginal distributions. And finally, we can show that in fact our parameterization can unify the 2 popular existing approaches, where the existing parameterization cannot do that. Under principal stratification, we are interested in estimating the average treatment effect among meaningful subpopulations, which will be termed as the principal causal effects. And we define the conditional probability of strata variable as principal score, and the propensity score as the conditional treatment propensity, also the conditional outcome regression. And to point identify the principal causal effects, we may also need other structural assumptions such as the treatment ignorability and the principal ignorability. In this project, we will use a popular statistical device, which is popular in the causal inference literature, that can motivate an optimal estimator in some sense. Based on the ERF, we can derive a doubly robust estimator. As the name suggests, this estimator is consistent and asymptotically normal on either correctly specified propensity score model or the outcome regression model, as long as the principal score model is correct. And the second, when all models are correct, this estimator is the semi-parametrically efficient, which means it has the lowest variance among a class of estimator. For inference, we will propose to using the robust sandwich variance estimator. ERF can also be combined with modern machine learners. In brief, we can estimate the nuisance functions, which means that the parameters that is not of interest, but we cannot bypass in estimation procedure. And we can show that many existing modern machine learners, such as random forest, neural networks, or superlearners, can be used to estimate the nuisance functions. We can show that under very mild regularity conditions, our estimator is as good as the doubly robust one, with our models correctly specified. And for inference, we propose to using a closed form variance estimator, which is computationally efficient. We conduct an extensive simulation experiments to investigate the finite sample performance of our proposed approaches. And this is the box plot of the bias in one scenario. The key takeaways are that incorrectly specified monotonicity may be dangerous in some scenarios, but misspecifying monotonicity as very large data odds ratio may only present a mild bias and under coverage. And we conclude with the future directions in this line of research. One can, for example, extend our framework to multiple categorical treatments using the concepts of high order odds ratios. And we also apply our approach to a critical care lung injury trial on the truncation by death, where 20% patients are dead in each arm. And in this trial, two active treatments were compared, so the monotonicity may not be an ideal choice. If you are interested in that, you can check out our preprint via this QR code. Finally, I would like to thank my advisor, Dr. Fanny, for his continued support, and Dr. Carla for sharing the initial research ideas with me. And that's all. Thank you.
Thank you. That was great, although obviously I did not understand all of your formulas. Well, I'll open it up to questions, just because I think each of you might have more sophisticated questions for one another. But also, maybe I could start by asking Zhangji, and this Xiaoxiao may want to respond as well. I mean, I think what's interesting as a practitioner, right, is to separate the sort of the adherence effect. Is it an effect that's inherent to the individual and their likelihood to take the medication, or is it an effect that's inherent to the medication itself because it's the lack of tolerability or some other sort of effect of the medication that makes people discontinue it? And so how both of you are proposing methods to sort of address this when evaluating the data. But how do you think about that or take that into effect? I'm just curious. And both or either of you, I'd be interested in your thoughts.
Well, I think the principal stratification can be applied when the patient, well, if you can, when patients take their treatment. For example, when treatment is signed a control arm, but patient is not willing to drop out the monetary benefit from the treatment that they would consider to take the treatment. So in this case, those patients are not compliant with the treatment assignment. and our framework is can work well on these cases.
Great. I don't know, Shajo, did you have any thoughts about that or not?
Yeah, sorry, maybe I feel like maybe I missed your question. Could you reframe it?
Well, what I was trying to get at, and I may have asked it in a kind of naive way, which is that it seemed like both of your methods are trying to address this issue of discontinuation and how to understand the true effect in that context with various approaches, if I'm understanding it, which I may not be. But so I was wondering how to disentangle the sort of the individual patient who's less likely to take the drug for lots of various reasons. Maybe their life is chaotic or there's other health-related issues that prevent them from taking the medications versus the effect of the medication itself, which leads people to discontinue because of lack of tolerability or whatnot. And if somebody can translate my question better, please do jump in.
I can understand. Maybe my mind was just absent. Yeah, that's a great point. I think you are talking about, the reason of dropout of the discontinuation. Sometimes, sometimes it's so in general, it can be classified into two types: the treatments associated and the treatment not associated, like you said, for some personal reasons, maybe life change or moving, and for the treatment associated, like the toxicity and the disease progression. Yeah, that's a great point. Actually, if we want to take deep into it, we can classify these intercurrent events into into two categories, like I talked, treatment associated and not associated. So we can use a different strategy, to tackle them if we want. And actually, there was one new publication, by my friends recently, dealing with such kind of problem. So that's a good point. So for us, we just for simplicity, we just classify the discontinuation due to all reasons into one general category, discontinuation. But a subclassification is beneficial. Yeah, definitely.
Thanks. And Shaja, if I could ask one other question that relates to your work, which is when you think about discontinuation and its relatedness to the medication itself, you were talking about you used a case study of a single trial, but do some investigators or have others thought about it as a treatment-wide effect and tried to use data from multiple trials to understand discontinuation effects as opposed to kind of trial by trial?
Well, we can. That's a good point. I mean, maybe we can extend our work to that framework. Yeah, but that's a great scientific question. I don't know how to deal with it right now. Yeah, but you just raised such a meaningful question. And
there's that one question from Cesar Torres.
Hi, yes, thanks. This is Cesar Torres from FDA. I'm a statistical reviewer, and I had a question for Xiaoxiao Zhou. Great presentation. This relates a lot to some of the work that I do at the FDA with respect to the principal stratum strategy. A large challenge with the use of the principal stratum strategy is the requirement of these assumptions, these underlying assumptions we do in the modeling, and they are strong and unverifiable assumptions. So within that context, it's very important to conduct these sensitivity analyses. And I saw on that last slide that you had in your presentation that you do plan to conduct these sensitivity analyses, and I think those will be very important. And I would go as far as to say that until those sensitivity analyses are conducted, it's a bit premature to draw any conclusions from the main analysis results, because we need to have a sense of how robust the main analysis results are to violations from those assumptions being made. So my question for you is, without having done those sensitivity analyses yet, do you have a sense, after having looked at the data, are you able to guess how robust the main analysis results are? How you expect the results to change as you vary those assumptions that you made in the main analyses?
Yeah, thank you. That's a great question. As you acknowledge that sensitivity analysis is an important part in the causal inference, since we assumed so many assumptions. Actually, we have done some sensitivity analysis in our work, but for time's sake, I didn't present here. And if you can recall my table of the proportion of each structure, I listed two results. One is the proportion of each structure with monotonicity assumption, and one is without monotonicity assumption. So in the first case, it just result in the 3 strata, and in the former, it leads to the 4 strata. So yeah, we have done that. And also for the weighting method, because we did it, we estimate in the Bayesian framework, and this assumption can be exposed, or you can do that without this assumption, like just using the general mixture model. So yeah, also in our package, you can assume or do not assume this assumption. So you can compare these results with different sites of assumptions easily from their results.
Great. There's a question in the chat from Tiffany to Alyssa. Tiffany, do you want to come off mute and ask your question?
Sounds good. Hi, everyone, and thanks all for all your presentations. Alyssa, thank you for your response to this. I'll go ahead and just read my question for the room. I asked, in your qualitative research, did you find that stakeholders generally felt similarly about the balance between rigor and the end in sight of data analysis? I was wondering if it was more polarizing or if you thought that folks were generally more on the same page about it.
Thanks so much, Tiffany, for your question and your kind words as well. And I'm going to basically reiterate what I put in the chat, that like they all felt similarly in terms of acknowledging the inherent tension in that balance. But the degree of that tension was, of course, different depending on the amount of trust that you had to put into the analysis and the decisions that you have to make based on that analysis. And if there's like legal and regulatory requirements that depend on the analysis or that are supported by the analysis in any way, then it required a lot more rigor and a lot more things needed to be agreed upon in order for the analysis to be considered complete and done than just a simple like ad hoc like A/B test in the software industry, for example.
Alyssa, I wonder if I could ask a sort of a follow-up question since you were just talking. The issues around registration and pre-specification and end of analysis, I mean, they come up a lot in observational research. They come up a lot, I can tell you, as a journal editor, when I wear that hat and I'm looking at articles, I'm sure it comes up a lot to the regulators too. One of the things that I've played with as an idea, and I'm curious what your response is the concept of triangulation, which is, in order to increase your confidence, because you never exactly know, I mean, there's timestamps and whatnot in terms of when people did their analysis and thus when they registered and pre-specified, but to see consistency in analyses across data sources, I'm wondering if that's come up in your work.
Yeah, definitely. And that came up a lot in the responses that I got during the qualitative interview study as well. And that's also part of like the quantitative methodology behind specification curve analysis. So it's definitely come up before, and it's an important thing to consider in all of these methods to make sure that the analyses, again, are as robust and replicable as possible.
Great. There's another question in the chat from Rohini to Shifeng. Rohini, do you want to come off mute and ask your question?
Yeah, thank you so much. I really love that presentation. I mean, all of them. I think I just completely may have missed this point, but since we are counting the number of events in the while alive analysis, is there like a way that the estimate in the end will help us differentiate, one group might be having events early on and another group might be having events later on in the same time frame, but the number of events might be the same in the estimate. So is there like, how can we pull that apart?
Yeah, actually, when we define the while life, we actually fix exactly time window to count the total event. So we actually provide a more generalized framework to estimate the treatment effect, regardless the early phase or the late phase. If you're interested in the early phase, we can fix the time at the early phase and count the cumulative event up to that time points. And if we're interested in the later phase, we can fix the later time points to count the total event. So we are able to compare the treatment effect at 2 different time points at the early phase or the late phase. So it's actually reduced and simplified to this framework to a fixed effect at exactly time window. So it's not a time varying effect, but a fixed treatment effect. But in terms of comparing the treatment effect at late phase and early phase, actually we're not able to do so because we assume that late phase people have more time to experience more events. So that's a not fair comparison because they exposed to the treatment longer in the late phase compared to the earlier phase. So we actually compare the treatment effect at a fixed time point, not late and early.
Jifang, if I can ask one quick follow-up before we have to end the session, but I'm curious whether the method also allows for weighting of the events within the composite.
Yes,
in particular, yeah, because sometimes there's events that are much more clinically meaningful, even beyond death. So okay.
Yes, we're able to do so. Yeah, and we are also able to, like, if we are not interested with the data at all, we can just assign 0 weights to that and just discard that.
Interesting. Great. Well, thank you to our 4 presenters in this session. Those were terrific. And we have scheduled a 15-minute break, and so we'll return at 1:45 for our final session. So please come back, and you can just turn off your video and mute, and then come back, and we'll restart 15 minutes.
I'll second that after. Yeah, he left a few minutes ago. I'm not sure where he is.
We give everybody 1 minute to get back to their seats. I'm looking at you, Michelle Bernabeu. Is Madigan on? Madigan's up first.
Yeah. Yes, do you want me to go ahead and start sharing?
Sure, why don't you do that?
Okay.
And then as soon as you're ready, we'll.
Okay, great. Okay, 1 second. Sorry, it's asking me to quit and reopen Zoom to share, so I might be out and right back in. Sorry about that.
No problem. Perfect. We're seeing speaker view.
Okay.
Yep.
Let me acknowledge that.
There you go.
Okay, great. So I guess I will go ahead and start if we're good. Okay, perfect. So hi, my name is Madigan Bedard. I am a postdoctoral fellow in Zoe McElliott's lab in the Bull Center for Alcohol Studies at UNC Chapel Hill. So I'm going to be telling you a little bit about some of our triangle CERSI work on the sex differences in responses to xylazine, fentanyl, and then the combination of those drugs in mouse models of withdrawal and respiration. So over the last 10 years or so, we've seen about a 10-fold increase in synthetic opioid overdoses. And this has been complicated by additives in the unregulated drug supply, really just like muddying the waters and making it really difficult to know what people are actually being exposed to and then how they might respond to different therapeutics or reversal medications. So we found that a collaboration between clinicians, public health officials, and preclinical researchers has been really crucial in our ability to understand and answer these questions and really understand the unregulated drug market. So you might have heard about xylazine. This is a substance that has started appearing in the East Coast drug supply. In the last few years, it's actually been around, especially in Puerto Rico, for about 20 years, though. And a few years ago, the Biden-Harris administration actually designated it as an emerging threat when combined with fentanyl. So it basically is an additional sedative that's being added, and it makes it really difficult to know how people are going to respond. Will they respond to naloxone? It also creates these wounds that are very difficult to treat and has generally just made the substance use field really more challenging to navigate. So it is typically known as an alpha-2 adrenergic receptor agonist, and it's a clonidine analog. And so you might have heard of clonidine, which is a common antihypertensive and has some other off-label uses, especially in treatment of opioid withdrawal. And xylazine is regularly used as a veterinary sedative in combination with ketamine, and it has actually no FDA-approved uses for human populations. So going into this project, this is really all we knew about xylazine because there was a lot of veterinary studies, but no studies on what this might do, in combination with other drugs or in people. So the first thing that we did, and this is actually the study that we brought to the FDA to initiate our CERSI funding, which was really exciting, was we looked at antagonist precipitated withdrawal. So this is a model our lab uses regularly. And in this model, we take mice and the mice get one injection, and the first injection is an agonist. And then we wait 2 hours and then they get a second injection. And that injection is going to be some form of antagonist. In the study I'm showing you today, it's naloxone, which is the opioid receptor antagonist that's commonly used to prevent overdose deaths. And then following that antagonist injection, I observed them for 10 minutes and scored their withdrawal behaviors. And those behaviors are kind of listed here. They are things like escape jumps, paw tremors, jaw tremors, wet dog shakes, abnormal posture, and fecal boli to measure, GI motility and those types of symptoms. So we repeat this for 3 days in a row, and the reason for that is that over those 3 days, we often see a sensitization of the withdrawal symptoms. So withdrawal gets more severe each time you go through it, which is a really interesting thing that we're specifically interested in modeling and then looking at neural changes following this paradigm. So in my study, I did something a little different than we typically do. Typically, I'm just giving morphine or fentanyl, but here I'm actually giving fentanyl, xylazine, the combination, or saline. So you can see that in this average Z-score metric, which is kind of a combination of those behaviors I previously described, we see that when animals are given saline and then naloxone, we do have some withdrawal behaviors, and that's not shocking. They're the lowest out of all of our groups, which is expected. We do have some, though, because we have endogenous opioid tone. So you have opioids in your body all the time. And so the naloxone is going to remove those from the receptors as well. Next, I looked at fentanyl, which is this purple line here. And you can see that in both sexes, the fentanyl followed by naloxone resulted in increased withdrawal scores compared to the saline group, and it seems to sensitize over the 3 days. When we looked at xylazine alone, and then precipitated with naloxone, we found that in the females, that looked really similar to fentanyl. We actually thought it was going to look more like saline because, as I mentioned, xylazine is an adrenergic agonist. So we immediately had the question of why is this adrenergic agonist responding to naloxone, which is an opioid antagonist? They're theoretically acting on different systems. That was really confusing, but also interesting to us. And we didn't see that to the same degree in the males. And then when we combined those two drugs and then administered naloxone, we saw that in females, you got this really dramatic sensitization of withdrawal over the 3 days, where that combination group is definitely the worse off. They're not feeling great by day 3, and it's pretty dramatic. In the males, you see that it tops out basically in the same place that the fentanyl naloxone group did. So because of these studies, we really wanted to look at what is the pharmacology of xylazine? We've always assumed that it's an adrenergic agonist, and that's what it does, and that's what it's been doing in the literature since the 1960s. So we partnered with XP Wong and Brian Roth and the PDSP, some other folks at UNC Chapel Hill, and actually looked into the pharmacology much more deeply than anyone previously had. And so this is a summary of those findings. So of course we found that it is indeed an agonist at the alpha-2 adrenergic receptors, but we also found that it is capable of binding to the sigma 1 and 2 receptors, which are, an interesting receptor that can act intracellularly to mediate, especially opioid signaling. And then most shocking, we found that it is actually a full agonist at the kappa opioid receptor. So when we looked into this and looked at our dose response curve, we found that xylazine has a PKI at the kappa opioid receptor of about 6.33. And then it actually has increased affinity when you look at each of its primary metabolites. So not only does xylazine bind this receptor, but so do its metabolites, giving us, an interesting new mechanism by which xylazine and fentanyl might have these interactions in the body, and why we might have been seeing that naloxone was able to cause withdrawal in the xylazine alone animals. So because we're thinking about how can we treat these people, how can we help them and prevent overdose, we want to know about the ability of antagonists to prevent or reverse respiratory depression. So this is really one of the next things that we've been working on. So in this study, we have mice and they go in this little chamber that everyone gets a saline injection, and we monitor their baseline, and then they're all given saline or an antagonist. And then we kind of want to see what does that antagonist do to respiration just on its own. And then we give an agonist, and in this case, I'm going to only show you a combination, which is 3.2 mg per kg of xylazine and 0.32 mg per kg of fentanyl. So pretty high concentrations of each drug, definitely higher than what I did in the withdrawal study. And then we look at the response over 90 minutes. So just as a brief introduction, I'm going to be showing you minute volume, which is a metric that we commonly talk about when we're talking about respiration. And it's really just a combination of breath rate, so how many breaths per minute, and then tidal volume, which is the volume in milliliters of air that is exchanged. So overall, we found that naloxone and atipamezole is more effective than naloxone alone at preventing respiratory depression that's caused by this specific dose combination. I want to, preface everything is like caveated by the fact that we're looking at, a single combination. And so the doses of each of these individual drugs could, of course, affect things. So when we look at just this combination of xylazine and fentanyl alone, we see that both in females and males in the gray line, we get about a 60% reduction in respiratory rate and minute volume. When we pre-treat with naloxone, though, in females, we see that it prevents that respiratory depression a little bit, but not to a huge amount. However, in the males, doesn't really seem to do anything. So this combination of drugs is clearly outcompeting the naloxone in this scenario. When we gave atipamezole alone in the red, we see that the females fared a little bit better, as did the males. But interestingly, when we combined those two drugs, it didn't change anything at all for the females compared to atipamezole, but we did see this additive effect in the males. So this kind of indicated to us that, especially in some individuals, there might be some benefit to combination therapies. Now, this bar graph is just kind of showing you a snapshot of what it looks like 15 minutes post-agonist injection, which is what we think is, the time to max effect. 15 minutes is about the peak effect for both xylazine and fentanyl. And so you can see some of the sex differences here broken out. So overall, I just want to emphasize that while these preclinical studies might not seem like they have great regulatory science implications, what we found from this project is it's really created a unique opportunity to create a pipeline of rapid translatability. So we are constantly talking with our public health collaborators and saying, what are you guys seeing on the streets? What new substances can we test? And so we're able to establish these assays and have faster response times and be able to, provide them with advice on things that they might be able to expect in these scenarios. Additionally, we've seen that we're able to talk with legislators and policymakers and be able to, inform them on what the current science is, and that can then help them influence decisions. And then finally, we see that xylazine really emphasizes the need for preclinical studies and then also preclinical studies in both males and females. So we've seen a lot of different sex differences here. And it's really important to say that both of those variables provide us with a spectrum of opportunity and kind of gives us insight into what is the possibility out there and how might different people respond because of sex or regardless of sex. So overall, I just want to thank, of course, my PI Zoe, and then my great undergrad Callista, who helped me run a lot of this, and our collaborators, and of course the Triangle CERSI for funding some of this. So thank you.
Thank you so much, Madigan. That was fantastic. Next up is Max Siebert from Harvard, who will be talking about implementing post-marketing requirements and commitments.
Thank you so much. Can you hear me well, and can you see my slides well?
Yes to both.
Perfect. Thank you so much. And thank you so much for the invitation and for the great presentations that we've heard so far. So I'm Max Siebert. I'm a fellow here at Harvard, and I'm going to present on the implementation of post-marketing requirements, leaving out the commitments for the sake of time. Okay. There we go. So before we start talking about post-marketing requirements, I want to take a step back and talk about pre-market evidence. And if you have listened to the talk and to Carla today, you know that clinical trial evidence supporting FDA approvals is getting lower, meaning that the number of pivotal trials are declining. However, that the use of expedited programs is rising, as well as the use of surrogate markers. And shout out to Yale at this point for doing such great work. So many people have said that the FDA has moved towards a model of increasing regulatory flexibility, and that products are approved on basis of greater evidentiary uncertainty in order to accelerate market availability. And so in order to counterbalance those uncertainties, the FDA has been granted authority to require post-marketing studies, and it can do so under 4 statutes. So there's the accelerated approval pathway. That was the first one, and it's also the most prominent one. There's the animal efficacy rule, the Pediatric Research Equity Act, and the latest edition, the FDA Amendments Act, or FDA. And so again, if you look into the literature, you will find that many studies are focusing, when it comes to post-marketing requirements, on accelerated approvals. And that makes a lot of sense because part of the accelerated approval post-marketing requirement is a confirmatory study to confirm efficacy. However, FDA FDA PMRs are the only ones that are focusing on safety, but all of the other 3, so PRIA, accelerated approval, and the animal efficacy rule are really focusing more on efficacy. This one is focusing on safety. And also important to keep in mind is that before this act, there was no obligation the FDA could not require a safety trial. And so what are those trials? And so I like this example a lot because it's on sampling and it's very famous at the moment, talked about a lot. And it is known that GLP-1 agonists have been associated with thyroid tumors. And so in this case, one of the post-marketing requirements issued under FDA was that the FDA requested a registry-based case series of at least 15 years duration to systematically monitor the annual incidence of thyroid carcinoma in the US. And so you might say, well, actually, this is great, right? There are checks and balances in place. So what is the issue? In 2016, there has been the report of the HHS Office of the Inspector General, which found that the FDA is indeed issuing more post-marketing requirements, but that there are challenges with its oversight. And so what are the objectives of our research? Well, we wanted to focus on post-marketing requirements that were issued under FDA. And our main goals were to track the status of the fulfillment, timeliness and completion, and also to capture labeling changes. So how did we approach that? We looked at novel drug approvals from 2016 onwards, and we extracted information on those from the FDA approval letters, as well as from the quarterly published and updated PMR and PMC database. So what are our results? In total, we found over 1,200 post-marketing requirements for 370 drugs. And this is for original indications as well as supplemental indications. And you can see that PMRs issued under FRDA make up to 2/3. And if you look at accelerated approvals, it's just under 10%. We were also interested to see if there is an association between expedited program use and PMRs. And if we look at drugs with at least one FRDA PMR, we see 253 or 68%. And we see that the proportion of the use of expedited program is higher, and that this difference is significant. However, we did this on a drug level. So we are interested to see if this significance is still valued when we do it on an indication level. But it would make sense, right? Because if you think that the use of an expedited program comes with some uncertainty and that this is balanced out by post-marketing requirements, it would make sense, would be interesting to confirm this. In terms of completion status, if we look at those that had a due date by December 31st, 2024, we see that 75% were completed. That's great, right? However, when we look at how many were completed on time, this number is shrinking to 31.6%. So in this graph, you see on the x-axis the time from the FDA drug approval, and on the y-axis the cumulative incidence of the study completion. And the orange line represents the planned completion date, and the blue the observed completion date. And the difference between those two represents the delay. And so in our case, this delay between observed and planned study time is significant. And if you look at the completion rates at 2 years, we see that there are 32.4% expected versus 23.5% observed completed, whereas at 5 years, the completion rate was 65.6% for the expected studies and 48.3% for the observed studies. Now, if we look at the label changes, here in this Sankey diagram, you see on the left the study types, and on the right, the different sections of the FDA label, the drug label. And you see where it goes. And so we saw that for 62.1% of fulfilled FDA PMRs resulted in a label change. And sometimes you could have more than one section modifications. This is where the ends are different. So which study type had the highest proportion of label modifications or associated with label modifications? This was PK/PD studies. And which sections had the most modifications? In our case, that was the use in specific populations and clinical pharmacology. So use in specific populations is, for example, people with hepatic impairment or renal impairment. And clinical pharmacology is usually pharmacodynamics and pharmacokinetics. Some of the challenges, so if you have already worked with post-marketing requirements, you might notice, but there is a lot of fragmented information out there that needs to be gathered together. Oftentimes submission dates are missing, meaning that documents that should have been sent to the sponsor and the timelines, these informations are missing. And then what you can do is you can send a FOIA request, but at the moment these are suspended. So that is one of the major challenges. So what are our key takeaways? We can see that PMRs issued under FDA are prevalent but remain understudied, that there are still issues with timely compliance, and that over half of fulfilled FDA PMRs lead to label changes. So our future outlook is to create a publicly available tool to track PMRs and also to explore avenues for policy impact and engagement with the FDA. So thank you so much, and shoot me an email if you have any question.
Thank you so much, Max. That was great. Next up, we have Mengyan Fu from Yale speaking on global impact of FDA drug approval, post-marketing label changes, and withdrawal determinations.
Thank you, Molly. Hi, everyone. I'm Mengyan Fu, a postdoctoral fellow at Yale. I'm very excited to be here today. And the whole project is about the global impact of FDA, including both pre and post-market decisions. And today I will share the design of our first study, which is drug and country characteristics associated with global access to FDA-approved novel drugs. So FDA is the world's leading regulator, with 2/3 of the novel drugs first approved by the FDA before any other global regulators. To avoid duplicative reviews and improve global drug access, especially in low and middle income countries with limited regulatory capacity, the WHO formally promoted Reliance Framework in 2021, which shift from independent national reviews to work sharing or even direct recognition of decisions made by other regulators. So among high-income countries, we can see bidirectional collaboration, such as Project Orbis for cancer drug simultaneous review. But more commonly, we see unidirectional flow of information from the leading regulators to the less resourced countries. In this scenario, although FDA does not regulate outside the US, and the global regulators maintain their independence in making final decision, the FDA approval often serves as a critical reference or may qualify a product for an abbreviated review process or even direct market recognition in other countries. So while reliance on FDA decisions help with the global access, there are growing concerns about FDA's increased adoption of expedited review. Between 2015 and 2022, 57% of the novel therapeutics used at least one FDA expedited pathway. For example, breakthrough and accelerated pathway allow preliminary clinical evidence and are associated with a 2-year shorter clinical development. These pathways do indeed facilitate earlier patient access to novel therapies, but they also introduce uncertainty of drug safety and efficacy to the global market. For other regulators where their post-market surveillance system are not as robust as FDA, they may easily follow FDA's approval decision. However, once a product is on the market, it becomes much more difficult for them to reverse the course. For example, the atezolizumab was approved for triple-negative breast cancer by FDA in 2019 through accelerated approval. Then after a fail in confirmatory trial in 2021, this drug indication was withdrawn in the US. However, only EMA mirrored FDA's decision. And this drug indication remained approved in 100 countries as of 2023. So we can see that FDA's regulation can sometimes have unforeseen impact beyond US border. So after our literature review, we found that most existing studies examine approval differences between 2 countries, often comparing with US FDA and focusing on specific drug classes, for example, cancer. A few reports have analyzed the global launch proportion and time lags for new drugs overall. There are also studies analyzed the association of country economic level with their launch of cancer drugs or essential medicines. However, there are no study analyzed the association of drug features and global launch and how different countries vary in their approved drugs. So our study poses 3 research questions. First one is what kind of new drugs have broader global diffusion? What kind of new drug have faster global diffusion? And how different countries consider novelty, clinical need, safety, and efficacy at their approval. In this study, we identify all novel drugs approved by FDA between 2008 and 2019, collect their characteristics, which has completed in our group's previous studies. For each drug, we will collect global approvals and launches as of 2024 from Setline from our projects. This is a commercial database, tracks global drug development activities in over 150 countries using public information. We will treat all variables as time invariants. For drug characteristics such as innovation pathway, safety, efficacy, we will assign values based on FDA initial approval. For country characteristics, we will use latest data from WHO and World Bank. We are still working on the data analysis method. Our current plan is to first use a generalized linear model to examine the association between drug characteristics and the number of global approval and launch. Then use a Cox proportion hazard model to examine the speed of global diffusion. And finally, use a logistic regression model to estimate the country-specific marginal effects for approval of different drug features. We conducted a pilot study using a small sample from our previous published work, which is 44 oral targeted cancer drugs first approved by the FDA between 2011 and 2018. And in our previous study, we calculated the use and spending of each sample drug in the US, stratified by evidence of benefit. And building on that, in the current pilot study, we found that other than US, these drugs had a median of 38 global approvals per drug. They have a median of 20 launches globally, covering 1.6 billion population per drug. From the country perspective, 75 countries were identified approved at least one sample drugs, with a median of 17 drugs approved per country. And 58 countries launched at least one sample drug, with a median of 13 drug launches per country. And among the 44 drugs, ruxolitinib had the highest number of global approvals, which is 60 countries. And enzalutamide had the highest number of launches, which is 44 countries. And pomalidomide had the lowest number of launch, which is only Canada. What's very interesting is that in our previous study, these three drugs were ranked third, fourth, and fifth among 44 drugs for annual total spending in the US. We are currently collecting full data and aim to complete the study in a couple of months. Our study has several limitations. First, our analysis is limited by the country coverage and available information in the database. And second, we treat all variables as time invariant, though they may change over time. Third, we lack data on manufacturers' global market application. So we cannot differentiate the regulatory delays and the non-submission. Finally, I would like to thank my supervisor, Joe, and the great team, Josh, Reshma, Harry, Sam, and Don from Peking University for support and guidance. And thank you all for listening today. We look forward to your feedback and suggestions. Thank you.
That was great. Thank you so much. And last but definitely not least, Tiffany Jiang from Yale on therapeutic benefit of the most expensive drugs covered by Medicare and Medicaid.
Thanks, everyone. Can you see my screen okay?
Yes, it looks great.
Great. Hi, everyone, and happy Friday afternoon. Thanks for your presentations. I'm happy to be here to chat a little bit about the therapeutic benefit of the most expensive drugs covered by Medicare and Medicaid. I'm a second-year medical student at Yale. I guess I'll start with a bit of background I think we're all familiar with. Drug pricing in the US is complex and harmful to patients. I'm sure we each come to the table with our own experiences with this, but the US healthcare system has among the highest prices of prescription drugs in the world. I'd like to draw your attention to this table. It shows that drugs are unaffordable for almost a third of the US population. This table was published in 2018, and I'd like you to focus on the highest rank, the US. We spend the most per capita on pharmaceuticals. For many common drugs for cholesterol, diabetes, asthma, US has the highest prices. Many new drugs do not necessarily show a clinical benefit above currently existing lines of care. Some research shows that among drugs with accelerated approval recently approved by the FDA, only maybe a third were recognized as having a high therapeutic rating. So this project sets out to answer, what is the therapeutic of our most expensive drugs? And there are several ways to measure therapeutic benefit. One metric that my group looked at was Prescribe International, which is a third-party French group, really unaffiliated with any governmental organizations, but independently publishes every quarter and rates drugs based on their clinical efficacy and a side effect profile, rating anywhere from Bravo all the way to unacceptable. Our friends who come from nations with health technology associations are probably familiar of HTAs. These are independent groups that give their own review of drugs and evaluate their benefit relative to currently existing lines of care. And these help inform government negotiations for drug prices. So here I highlight the group in France and a group in Germany. And just an example of their rating scores, a major rating of 1 would suggest that this is a major improvement of clinical added value. So I set out to answer, what is the therapeutic benefit of the 50 most expensive drugs as measured by cost per dose covered by both Medicare and Medicaid? This was a descriptive cross-sectional study that uses the CMS spending dashboard. You can Google it, it pops right up. And my variables of interest were years since FDA approval, their indications, and if they have any orphan drug indications, and level of innovation. This is sort of based on a first-in-class designation from CDER. Safety and effectiveness from Prescribe, as described, and the added therapeutic benefit rating from the HTAs previously described. This is an overview of the 50 drugs. You'll see that the median cost per dosage in 2022 was $16,000. Majority of these drugs were biologics. A quarter of them had immunologic indications. A quarter had oncology-related indications. And the majority of them had at least 1 orphan drug designation. I'll give a moment just to parse this graph. This is the top 10 most expensive drugs by dosage. These will be pretty familiar to y'all. We have anything from gene therapies to CAR-T immunotherapies, all the way to antibodies and recombinant proteins. Average spending per dosage unit ranges from 1.3 million for gene therapy to 40K for the top 10 most expensive drugs. And only a handful of these most expensive drugs had what we consider high added therapeutic benefit. Overall, only about a third of them received at least 1 rating either from the French or German HTAs of having high benefit, and more than half were rated as having low benefit, and the remainder were not reviewed or withdrawn from the EU. From the prescriber side, only 1 drug, Givlari, which is a siRNA indicated for acute hepatic porphyria, was recognized by prescriber as being either Bravo or Real Advance. And of these 50 drugs, 6 were labeled as unacceptable or outright advocate to avoid because they're adverse event efficacy profiles. So we'll walk through a few takeaways from this initial study. First, few of our most expensive drugs have high added therapeutic benefit as measured by HTAs in other countries. Many drugs were approved for use, for which manufacturers then set very high prices. However, the majority are determined to be of low therapeutic benefit, while few are determined to be of high therapeutic benefit. And I think this is a good opportunity for some discussion here, because obviously drugs are incredible. This is how we can make medicine better. Many of these drugs are entirely how we change patient lives and revolutionize how we manage disease. At the same time, many patients cannot afford their medications, and drug prices in the US are time and time again higher than in other countries. I want to add for context that this study looks at the cost to Medicare and Medicaid, which is separate from patient out-of-pocket costs. Countries like Germany and France negotiate drug prices with these pharmaceutical companies, and these HTA clinical benefit assessments can help decide pricing and coverage. Continuing this study, most of these expensive drugs are now eligible for Inflation Reduction Act price negotiations. Of our 50 most expensive drugs, only 2 would be likely eligible for the next round of negotiations. This would be aflibercept and adalimumab. There are several reasons for exclusion from or current exclusion criteria under the IRA, ranging from anywhere from they're covered by Medicaid and not Medicare, or they are approved relatively recently, or they have a single orphan drug designation. The second takeaway is that almost none of these drugs are eligible for IRA price negotiations under current exclusion criteria. And so we kind of sit here with this cross-sectional descriptive study and wonder what's next? What can we do? I think at the policy level, I think that we are aware that the FDA has recognized the importance of affordable prescription drugs as a public health issue, as access to medications significantly impacts health outcomes. Policies are needed to better align drug prices with therapeutic benefits, including perhaps considering broader eligibility criteria for Medicare price negotiations. And because we saw that so many of these were biosimilars, considered that we could have lowered barriers to market entry. There are a few policy notes of interest here. I'm sure y'all are familiar that in 2023, part of the Inflation Reduction Act was to help with insulin affordability. Under this policy, under Medicare, insulin would now be capped at $35 per month under Medicare Part D. So this is an example of how policy can directly influence drug costs to Americans. And another side of policy, kind of holding industry accountable. FTC last few years had a history of subpoenaing a lot of industries and recognizing when there are pay-for-delay schemes. These are ways that drug companies can enforce their monopolies by delaying competitors from entering market. And of course, there's at the patient level, where it's not enough to just care for patients. It is important to check in with patients and ask if they're able to afford their medications, and helping advocate either through repealing insurance processes or helping them find payment programs or find other lines of care so that they can actually get the care they need. I want to give many thanks to the CRIT team and CERSI, and to thank you all for listening, and looking forward to chatting more in the Q&A.
It's fantastic. What a great panel. I really enjoyed every one of these presentations. Does anyone have any pressing questions? Otherwise, I have a question for Matt again. So I'm so curious about this unexpected kappa opioid receptor thing, which had not previously been reported. The thing that I find really fascinating, and you kind of alluded to this, is like, okay, so it starts showing up in our drug supply. And I guess I kind of assumed it was just because it's somehow cheaper or easier to import than fentanyl and has a similar sort of depressant effect. But is it that people who use drugs actually have like identified this, even though pharmacologists hadn't?
So that's really interesting. So there's a bunch of different theories about why xylazine really started appearing. The consensus is that most people don't like it. One of the theories, though, is that, people, if they had their drug of choice, it would be heroin. And with the rise of fentanyl, heroin became almost impossible to find. It's now kind of making a comeback. But one of the theories is that dealers and chemists were trying to make the fentanyl feel more like heroin because the fentanyl is such fast on-off effects. One of the theories is that they were thinking, oh, well, the xylazine also being a sedative kind of extends the high, extends the effects, which doesn't necessarily seem to be the case. It just seems to just make people unconscious in most cases. But there are a small subset of people that do really enjoy xylazine and seek it out. But yeah, it's kind of an interesting situation because people have just such varied responses to it.
Interesting. And just as a follow-up, like how close, you mentioned, this is very bench sciencey. How close do you think you are to treatment recommendations for xylazine fentanyl overdoses?
So our primary goal is to answer the questions that the public health officials have and that first line responders have. So we are, running the mouse assays that we can run to try to inform those studies. But the biggest thing that we set out to do was look and say, is naloxone going to be effective? Because especially a few years ago when this really started ravaging cities, there was some talk of people saying, well, if you think xylazine is involved, don't give naloxone. And so our first goal was really to say, no, please give naloxone. It might make withdrawal worse, but it's not going to hurt in terms of respiratory depression. It might help, it might not. Might be some subset of the people that it will help in. We might need more than that. So that's kind of where we started. And I don't know exactly how far away we are from being able to, really advise treatment recommendations, things like that. But our goal is to establish this pipeline so that as new things come into the drug supply, which is happening all the time, the big one is dexmedetomidine right now, which is another alpha-2 adrenergic agonist. We're able to more quickly answer some of those really basic questions that users, harm reductionists, public health researchers all have. So that's kind of where we're at right now.
Great, thank you.
Yeah.
We have a question in the chat from Irfan. Do you want to ask or do you want me to read it? Please feel free to come.
No difference. Thanks all for the presentation. I had a question. In Tiffany's presentation, I saw that like 90 out of the 50 drugs didn't have a specified therapeutic category. I was thinking maybe a better classification could be better to be used for categorizing the therapeutic area of these medications.
Gotcha. Thanks, Irfan, for your comment. I want to say that's a great point. Others seem to be a way for me to lump in anywhere from ophthalmologic to endocrine conditions. And I think that's a good point. There's a pretty wide spectrum of indications for these drugs.
Oh, you classified the drugs based on the indications?
I believe, are you referring to a table in which under other, this referred to their indications?
I mean, there was a slide. There were like some rows, cancer or other types. There's a classification, ATC classification that like classified the drugs based on like their pharmacology class. Maybe you can use that for classification of the medication in your study.
Sure. Thanks for your comment. To that point, yes, there's pharmacologic class is possible. I think that could be broken down under like biologics or small molecules. Oh, thank you, Irfan. Yes, that sounds good. Both indications and ATC.
Christine. Oh.
Sorry, getting my act together here. I have another question for Madigan, and I'm sort of cheating because I know a little bit, a lot more about Madigan's work since she's part of our CERSI than I do about the others. But I thought that there was a pretty important point you made at the end of your presentation and on this summary slide about the importance of the females being included in the model. And I know that we're all facing a little bit of uncertainty about using females or I guess the loss of female sex as a biological variable in our upcoming research. And I know that's sort of an issue, something that you guys are taking on a little bit with some passion in your lab. And I just wondered if you had any suggestions for this community as to what we could do together to kind of like continue to advocate for how important it is to continue the role.
Absolutely. Thank you, Kristen. It's incredibly important. I mean, I think that if you look at my data and you look at what we see in the males, it looks like the combination of xylazine and fentanyl is no worse than fentanyl alone. And then you see the female data where suddenly this combination is drastically worse than either substance alone in those female mice. And that's just one of many examples we have. But I think that as we start to get we are prioritizing how can we minimize animal numbers, how can we be as efficient as possible with animal research and preclinical studies in general. And so an easy suggestion a lot of people have is like, okay, well, just pick one sex or the other and study that in whatever you think is most relevant. But I think this really exemplifies how one sex or the other is not more relevant in this scenario. We need to understand all of it. And so if you're trying to focus on, potentially decreasing numbers, we pilot everything in both sexes at lower ends. And then, if we're not seeing sex differences, then you can collapse across sexes, of course, and proceed that way. But I think that this really is just a testament to how important it is. And we hear anecdotally from people in the clinic that, a couple will purchase fentanyl that has xylazine in it, and they'll both use it together, and the husband will be passed out on the couch for 3 days, and the wife is hyperactive and like feels great and loves it. So we do just see such varied responses, and I think it's such a crucial variable that we cannot ignore.
Thanks. Go ahead, Mary or Corey.
I also had a question for Madigan. Amazing presentation, by the way. I was just wondering whether there are any evidence in the literature for race or ancestry-specific differences in the xylazine and fentanyl combination response that you see, as well as I'm curious about your hypothesis of whether that is those differences in the sex difference that you saw come from the genetic variability or more environmental variability between the sexes.
Yeah, both really great questions. So the short answer is no, there's almost no literature on any of this, especially with the combination of the two. I have some suspicions that there might be, genetic and racial components to some of these things, and to the effects or responses to some of these drugs, just in some of the things we see with clonidine, because there is more literature on clonidine, and there's a few case reports out there where there's adverse effects in Black males, but not in other populations, and things like that. So I think it's not ruled out by any means, but there's not enough to really say much on it. And then to your second point, I think so the strain that we're using in the withdrawal study is a very inbred strain, so they're very genetically similar. So I think it's definitely probably hormonal estrus cycle related, but probably, which is, definitely something we could investigate more with, and more carefully with the different methods we have to look at that in the lab. We are doing a lot of our work now in a more outbred strain, and we're still seeing a lot of sex differences. So it's kind of just a whole mix of things right now. We just don't know enough. And so there's so much more work that's needed, but those are really interesting avenues of thought.
Super interesting. Thank you so much.
Yeah.
I have a question for Mangyan. So I guess I have not really thought a ton about how the different drug agencies around the world sort of are interacting with each other. And I was really curious, from one of your pie charts, it said that the FDA was first in, it was like more than half of the drugs, first in the world. And then Tiffany mentioned, okay, but we also have the highest drug prices. I'm just curious if you have any thoughts about like how sponsors are sort of phasing where they go in different countries. Do they hit them all at once? Are other countries sort of are the sponsors choosing to go different places versus the countries are sort of waiting to see what the FDA does? I'm just curious about kind of how that works.
Oh, thank you, Molly, for your question. That is a very interesting question. So from the manufacturer's perspective, right, whether they choose to submit their application in some countries or whether they choose to launch their drugs in some countries are actually in the black box. Nobody knows their strategies. And there are also not specific data for, you cannot track their preference in, submitting to the application worldwide. But there's a thing is that, so for example, Latin America, the researchers there are very worried about because, documented in their policy, it says that, oh, all the FDA approved drugs are very welcome to come to the Latin America countries. We can give you a lot of abbreviated approval or recognition. However, when you see the real launch proportion of the drugs there in the Latin America, the launch proportion is very, very low. And we guess it might be because of the manufacturer choose not to launch there because they have small markets. The population are not very large. They cannot afford the high price of the new drugs, reasons like that. So this is very interesting. We can do maybe survey or interview with the stakeholders to learn more about it, what they're thinking about. Thank you.
Thank you. Question from Philip.
Hi, this is a question for Max and a comment as well. Max, great job. Really enjoyed your presentation. I actually think, and I should give some context, I'm a physician reviewer at FDA at CBER, the Center for Biologics Evaluation and Research. And basically your presentation was, it was so good that it could be, for our group in particular, I think it could be a nice primer, especially for our younger newer reviewers who could use the context that you provided as to why we do what we do. We basically work with the offices during the process of approval for vaccines and biologics. And then, like you said, if there is a certain level of uncertainty, we're kind of like the consultants who issue the post-marketing requirement notification to the manufacturers and then follow up on how they follow up whatever concerns that we have through post-marketing requirement studies and other actions. So definitely a great fit in terms of our group and really well presented. I was just wondering if you or your team had thoughts on or if you had any thoughts on what could be attributed to why the compliance wasn't like 100%, like if you had any thoughts as to, the reasons why companies weren't meeting those goals to complete their PMRs on time.
Thank you so much, Philip. And great work that you're doing. Happy to see that there are some people behind those efforts. It's different, right, for every kind of post-marketing requirements. For example, we're seeing for PRIA, we're looking into that as well, and there we're seeing kind of delays and deferrals. You're right for post-marketing requirements. Sometimes you have explanations in the database. For example, patient recruitment is taking longer, or sometimes you can also have a modification of the post-marketing requirement, especially when it comes to safety, for example. Sometimes things change, and that can, for example, lead to a release of the post-marketing requirement, but also to a modification and then to a to something different. So, but I guess that's a fair point. I think it would be interesting to see what are the delays and also what are and how to improve that. And I think maybe there need to be better talks with the sponsors and the FDA, right? Because they are discussing the timelines beforehand. But maybe the FDA is setting unrealistic timelines. This is also another, right? It's sometimes also a black box for us. So definitely something worthwhile looking into. Thank you for your comment.
Thank you, Max. Yeah, I can definitely attest to the reasons that you gave on an individual, like file to file basis, are reasons why the milestones, we call them, are not met on time. I think what's interesting in terms of what you have to offer is, and what we would not maybe readily know based on our day-to-day work, is that aggregate picture of why sponsors tend to not submit on time. So I think that's what's interesting about your work, is seeing it from, that high level as to what the reasons are. But again, great job. And thank you for your contribution and your interest in this work. Appreciate it. Thank you.
Thank you.
Thank you. We have 3 minutes left, and I think that's absolutely the perfect place to leave it. So I will just give some very quick closing remarks to thank all of you for joining today for our annual CERSI Scholar Virtual Research Seminar. As always, it was such a pleasure to hear all of these presentations from a group of scholars addressing questions across the entire spectrum of questions that are of interest to the FDA and of interest to regulatory scientists worldwide. Thank you to everyone who joined today from the FDA, our scholars and mentors and friends of the CERSI, and from other institutions, regulatory science groups. Thank you to Dr. Kozlowski for his opening remarks. The CERSI Scholars program is an important part of our mission as the Yale-Mayo CERSI, and we're really seeking to develop future regulatory scientists and to support the science that they're doing that answers essential questions that help the FDA in its work. And last year we heard, and I had not heard this before, but the FDA's work regulates products making up 25% of all consumer dollar spend in the United States. Such important work. The scholars who presented today, your projects covered a huge variety of questions, right? I mean, we had biostatistics, we had bench science, we had regulatory policy, we had meta science. And all of you in different ways were really speaking to the opening remarks that we had today on disruptive innovation and the role of the FDA in making innovation possible to ultimately support the health and well-being of every single American. And it is just such an honor to be part of this incredibly important endeavor. I want to say thank you to all of you for the work that you do. Lastly, a big thank you to Michelle Bernabeo for her work to coordinate the program today. Thank you, and we look forward to seeing you all back here next year. Thank you all.
Thanks, everyone.